2:45. Yeah, that's to 3 o'clock. And then content block 3 will run at 3:15 to 4:40 to 4 o'clock, I think. Yeah, that sounds right. I did 3 maths and I can't even calculate 45 minutes. Anyway, that's all right. In content block 1, so in the first 45-minute block, what are we going to be doing? I'm going to start off with a bit of VCE sort of bio-specific advice, because I think that always pops up in these types of formats where I get messages saying, hey, Michael, what do you recommend? How many hours a day should I study? What should I be doing to do well in bio? How can I get a 45 and above? And that's cool, right? I had those questions too when I was back in VCE. So I just want to give you a few, a bit of, general tips to help you guys succeed, because I think that's really important to cover. And then we're going to get started straight into Unit 4, Area Study 1. So for some of you guys may have already covered Area Study 1 at school and are pretty much, about to finish the first half of Unit 4. Some of you may have not even touched Unit 4 yet, but nonetheless, I still want to go through this, and as I'll say in a moment, I recommend that all of you sort of try to get ahead with your schoolwork and try sort of push forward and cover as much as you can of Area Study 1 content in Unit 4, because getting ahead with this stuff I think is really, really key with VCE. So that's about what I've got to say about the first 3 content blocks. And then, content block 2, we continue with Area Study 1, and then content block 3, we may get time to go through Area Study 2, but quite frankly, I've got a whole bunch of slides today, so if we don't finish, that's okay. Quite frankly, I'm not planning to finish all the slides I've got, because obviously my philosophy when teaching is I'd rather teach something a bit more slowly and make sure that you guys actually understand what I'm saying, as opposed to just rushing through all my slides and then you guys coming out of this 3-hour session coming out knowing nothing, right? So I'm going to try to take it a bit slow and hope to at least cover all of Area Study 1. And then there are going to be notes that you guys can download and you can, read up on Area Study 2 if you wish. Now, another thing before I get started, and this is probably the last thing before we actually get started with content, is just a few notes, right? So in terms of these lecture slides that I'm using today, I'm aware that you guys, on the same website that you guys are actually watching this live stream, if you just scroll down a little bit, and I think it's on like the right-hand side of where you guys scroll down, there should be sort of a link or some sort of like file that you can download, and that will be these lecture slides. It says there are slight modifications. My ones just have the solutions to the questions which we're going to go through in today's lecture. Yours obviously will not, because that would defeat the purpose. So that's one sort of difference. And then the recording what you're watching this video is going to be recorded, and you guys can actually watch that recording again on the same website that you're using to actually watch me right now. So if you forget to take note of something, or I said something that you remember now but are going to forget in a week's time, it's all chill. You can see this recording again in whenever it's available and whenever it sort of uploads, right? So don't stress, right? It's all chill. Alrighty, so let's get started with some VC bio advice. What should you be doing during these holidays? Really classic question, and this is my biggest advice, right? So try not to, bludge too much, because quite frankly, what type of holiday is this? Number one, we're in lockdown. It's not really a holiday. It's a bit depressing, but that's okay. But, you guys, I hate to bring it up, but the ATAR is a competition, right? It's a rank. So you have to be thinking, what is everyone else doing to get ahead? And as a result, please don't use these, 2, 3, or whatever they've extended the holidays to for you guys, obviously you need a break. But try to get ahead with some of your schoolwork too. So what I particularly recommend is try get ahead with Unit 4. So when I was doing VCE Bio, the thing that I think helped, or one of the things which I think helped the most, was trying to get ahead with the content by studying on my own. So making my own notes from the textbook and reading up on the textbook before I did it at school. And that way I could actually treat school as a bit like revision for what I'd already studied, by myself, right? And if anything, if I missed out on any content or didn't quite catch something that was important in the textbook that was, really reinforced by my teacher or by my school, then that's what I'd use school for. I was also a bit of a chatter person, right? So I'd also, just bludge a lot in the classroom and just chat to my mates. So it also meant that at home was when I did my study. At school was a bit more sort of social in that way. And then in addition to getting ahead with Unit 4, I'd really, really, really recommend that you guys don't forget about Unit 3, right? It's a really classic thing that people, you guys just finished Unit 3 or you might be just about finished with Unit 3 and then you don't touch it until you finish Unit 4. Right? And by the time you finish Unit 4, it might be, the end of August, early mid-September. And by all that time, if you have not touched Unit 3, I can assure you that you're going to forget all the little details that is really important to score well. So whether it's remembering what exactly the T helper cell does in immunology or remembering how exactly transcription works, you don't want to waste time, in the lead up to your exam, relearning that content. You want to go straight into practice exams and smash those out. So the best way I think you can revise is just set maybe an hour, 2 hours a week or a fortnight to just focus on revising some of the older stuff that you would have covered in Unit 3. And another great way to do that is because you've learned the content, just do some practice questions. So I'll give you a few tips into where you can get some practice questions later on, but I'd highly recommend, any types of sort of publications. Like I know we've got, our atarnotes topic tests, and there are other sort of resources that you can use to do that too. So I'll touch on that point a bit later on in this slide. And then what other tips can I recommend for you guys just in general? Right, the first one is to make your own notes, right? It's a bit of a no-brainer, but sometimes I come into contact with students that I'm teaching that sort of say, oh yeah, I'm just going to use your notes, or I'm just going to use, this company's notes. And it's like, yeah, that's okay, but don't be lazy, right? Try and make your own, because if you don't make your own notes, you're not really going to learn the content properly. So yeah, that's one big thing. And then when making your own notes, try to spam it with, diagrams and colors, and don't make it just big blocks of text, because as soon as you do that, I don't know about you guys, but when I'm reading big blocks of text, I might, get to the bottom of the page and then I just think, what the hell did I just read? I was about to swear for a moment. But yeah, you think, what on earth did I just read? It just went through one ear out the other. So for me personally, by looking at sort of pictures and other visuals and that sort of stuff, I sort of think about the content a bit more and I'm more engaged with my notes, and that way I sort of take a bit more from my notes as opposed to just reading big slabs of content. In addition, do practice questions. That's my biggest advice today, right? I always get questions in these lectures being like, Michael, how do I make my responses to short answer questions better? How do I write more concisely? How can I do this? How can I do that? And honestly, it all stems back to just doing practice questions, right? There's no specific formula or secret or whatever to writing a good short answer. It really is from practice. Because by doing particularly practice VCAR questions and then looking at VCAR examiner reports, you then get a bit of an idea about, okay, VCAR sort of wants or expects this point, and this point when talking about, a particular topic, such as apoptosis. And so by getting exposure to practice questions, you start to develop a bit of an idea about, what you need to write for your exam. And that's probably the best way to fix up your short answers. And I know for me personally, when doing my SACs at school, I was sort of the type of person who would write essay-long responses for, your 2 markers or your sort of small questions, because I was just a bit extra like that. But then it was only until I started doing practice questions and doing particularly VCAR practice questions and looking at their sort of model responses, where I sort of developed a bit of idea about how to cut down on my responses, what was the important bits, what were the sort of superfluous bits that I was sort of just rambling on about, and then fine-tuning my responses so that I was actually doing a good, or at least a better job when answering questions in the short answer. Rightio. The next one, and this is probably something which I think I could have done if I wanted to do a bit better in bio, but I'm happy with what I got. But focusing on your weaknesses is so important, right? This might, not be such as a big sort of tip right now, but when doing your practice questions on practice exams in, the next month or two, you'll begin to realize that, you'll love doing questions on a particular topic that you're confident with, right? So for me, that was Unit 3. Absolutely loved Unit 3. It was something which, when I started, I was getting, high 80s, low 90s, and then just worked my way up to mid to high 90s on doing these practice exams. I absolutely loved it, right? I just understood it, and it's something I'm really passionate about. But then back when I was in year 11, it's changed now, but back when I was in year 11, I wasn't a big fan of Unit 4, right? I really liked the cell stuff and the molecular stuff, and I wasn't a big fan of the whole, evolution of big picture sort of perspective. And so when doing Unit 4 stuff, I just didn't enjoy it. I thought, nah, I'm not going to do questions on this. It's just not something I like, because I didn't like getting not great or, marks which I wasn't impressed with. But then honestly, you need to focus on that. And so I pushed myself, and I was able to work my way up from getting, let's say, I think it was about 70s when I first started practice exams, Unit 4 questions, to then up to 90s when doing practice exams for Unit 4. So again, you need to focus on your weaknesses, because quite frankly, VCAT doesn't care if you're not good at a particular topic or not good at a particular concept, right? They're going to test it. So you better cover your bases and work on your weaknesses, because that's the biggest advice that anyone who gets, in the 90s for their ATAR, or who gets above 40 in study score. That's probably the biggest advice that you're going to hear from anyone that gets that high. So really, really, really do focus on areas of weakness. And then finally, a bit of a wholesome point, but really focus on your well-being too during these times. Obviously, everything that, it's a bit of a cliche to be like, oh, it's unprecedented times. But it is, right? No one's ever gone through this. No other Year 12 or Year 11 cohort has ever gone through what you guys are going through at the moment. So I really just want to reinforce the fact that, take care of yourselves, make sure that you're doing okay, because that's really important to do well at school. So whether it's, making sure that you're taking, study breaks and doing some exercise or going to bed earlier, and making sure you get, your 8 hours of sleep or whatever, amount of hours of sleep that you sort of work best with. I really, it's something which you guys might be like, Michael, hurry up, I want to get into the bio. But it's important, right? So I know with me personally, I was terrible with sleep and that sort of thing. But it was only until recently with this year uni and a bit of last year uni where I thought, I'm going to start going to bed a bit earlier, at a reasonable time, wake up, and get my 8 hours of sleep. And it's incredible what it does. When you start taking care of yourself, it's really incredible the impacts it has on then your marks and sort of your overall sort of well-being. So 100% focus not just on study, because no one who gets, a high or good score just studies, right? You need to focus on your well-being too. So really, 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 jokes aside, make sure that you're looking after yourself in this tough time. Chat to your friends, do all that sort of stuff, because it's important. Rightio. That's all the advice done. We'll go through some more questions as well. Something I probably should have mentioned, right? As you're seeing me on the same website, if you just scroll down, you'll notice that there's a little chat box sort of function thing where you can post any questions that you want to ask. At about 10 minutes or so, I will go through some of those questions for about 5 minutes, and we'll do that every single sort of content block. At the end of each content block, I'll go through and run through some of your questions. Alrighty, let's get through some content. Right, so we're going to start off with looking at sort of genetics and the involvement of genetics and its influence in affecting how we look, right? And that's going to be pretty important when considering things like natural selection, speciation, things like evolution, which I'll talk about in the next few slides. So before I begin, I obviously need to, introduce some of these terms which you may have come across before, but it's important, right? Otherwise you won't understand what I'm going to be saying in the next few slides. So the first one is gene, right? Everyone should know what a gene is from Unit 3. A gene is just a bit of DNA that, for VC purposes, is a bit of DNA that codes for RNA, right? And then an allele, right, you might remember from Units 1 and 2, is a variation of a gene, right? So for example, you might have an allele that codes for blue eyes, and then you might have an allele that codes for brown eyes, right? So there's one gene, a gene that codes for eye color, but then there are different variants of that one gene that code or that produces a different sort of variation of what that, actually ends up looking like. So, you might remember like the big B for brown eyes and the little b for blue eyes, right? They are alleles for the single, gene locus. And then we can sort of say that the gene pool, right, it's the total collection or the total sum of alleles that are found in the population, right? And so the more alleles we have, you can say that there's a larger gene pool, that there's greater genetic diversity in this population. And then finally, the phenotype is effectively what the expression of the gene ends up being, right? So if the person has blue eyes or green eyes or brown eyes, right? Or if they've got black hair or blonde hair, whatever it may be, it's sort of that physical, biochemical, or, physiological sort of expression or, characteristic or trait that comes from, the interaction of the person's genetics as well as their environment as well. Right, but for VCE purposes, we don't really talk too much about the environment's influence. Alrighty, so with that said, right, we're going to start talking about mutations. And the first thing I want to mention is the fact that when we talk about a mutation, we're talking about a change in the DNA of a particular organism, right? And that's sort of represented by a change of a nucleotide or a series of nucleotides. Now, when we think about mutations, right, we sort of think about really bad things, right? When you think of mutations, you might think of, radiation and things. You might think about things like cancer, which is, related. You might think about, these sort of like negative, stereotypes, if that's the word. But not all mutations are bad, right? What's important to understand is that mutations, as we've just mentioned, they change the genes in our DNA, and as a result, they are the source of new alleles, right? They're creating new genetic variants in our body, which gives rise to different traits, or new traits, I should say. By giving rise to new traits, these could either be really good, or they might be really bad. But the point is, it's completely random. We don't really know what's going to happen, right? But just understand that mutations, they're the source of, new sort of characteristics that we find in an organism, and they can either be good or bad, right? And that's going to be important in a moment. Now, mutations, right, they can occur in two main areas. We could get what we call a somatic mutation or a germline mutation. When we talk about a somatic mutation, we're talking about mutations which affect any type of cell that doesn't produce gametes, right? So I'm talking about things like skin cells, our muscle cells, neurons, whatever it may be, any type of cell that doesn't give rise to, sperm or eggs, right? Because the special thing about somatic mutations is that they can't be inherited, right? If you've got a mutation in a skin cell, for example, that change in the genetic, makeup of that cell isn't then going to get transferred to your gametes, right? Your sex cells, because, your skin cells have nothing to do with the cells that end up being, the sperm and the egg. So the big thing is that's not inherited. So for example, if someone gets a skin cancer, it doesn't necessarily mean that their children have a higher chance of getting a skin cancer, right? Assuming it's a mutation that just happened in their skin cell or in their somatic cell. However, that changes if we're talking about germline mutations. So if there's a mutation in the DNA of your actual, germline cells, which are the cells which go to make and produce the gametes, the sperm and egg cells, then of course that's then going to get transferred into the offspring, into the children of that person. So that will be inherited. Right, and now when talking about particular mutations, these source of new alleles, right, there are particular types of mutations we can talk about. The first type of mutations we're going to be talking about are substitution mutations. And this is where we effectively take one nucleotide and swap it out with another nucleotide, right? And there are three main things that can happen from doing this. Looking at the picture, my cursor doesn't seem to be working, I'll use the pen. So looking at the picture, okay, that's all working, that's good. Right, our first type of mutation is what we call a silent mutation. What happens here is that we get one nucleotide, so we get one base, and swap it out for another base. In doing that, that triplet, which then goes to form a codon, produces the same amino acid. right? There's no change in the resultant protein of that gene. And you can sort of see that here, right? So what it's saying is that in the triplet of the DNA sequence, what's happened is that this A has been swapped out for the T. In making that change, right, GTA to GTT, right, that has changed, or I guess I should say that has not changed the resultant amino acid, right? We're still producing valine at the end of the day. As a result, we call this a silent mutation, and I like to think about it because, I like to think about it by thinking it's a silent mutation because you don't see an effect, right? There is no change to the protein, so it's silent. The next type of mutation is what we call a missense mutation. What happens here, as we can see in the image, is that this C has been swapped out with an A. So instead of having the triplet CCC, we have ACC, right? Sort of like the sports sort of competition that some schools do. Anyway, so CCC to ACC, right? What that does now is it actually changes the resultant amino acid. We go from proline to threonine. By changing that amino acid, it obviously is going to change the protein, right? So I like to think about it as, missense is being misplaced, right? We're misplacing the amino acid by changing the nucleotides here. That's how I like to remember it. So missense, it's misplacing the amino acid, so you've changed the amino acid. And then finally, we've got our nonsense mutation, right? And what happens here is that looking at this, you've got TAC gets converted to TAG, right? So by changing the C to the G, we've changed this amino acid, tyrosine, into a stop codon. And as you guys are aware, stop codon is going to cut this protein short, and that's going to, therefore stop the production or the addition of any other sort of amino acids that come after this triplet. So because of that, the protein is going to be non-existent, right? Because it's a stop codon. That's the way I like to remember it. It's a bit stupid, but if that works for you, it works for you. The next type of mutation, or the next type of substitution not substitution mutation. The next type of mutation is what we call a frame shift mutation. So here, we're not swapping out anything. We're adding or deleting an amino acid, a nucleotide from the DNA. So what you can see here is this. What can we see? We have, I'm just looking at the differences here. For some reason I can't see the difference. TAT. Anyway, I can't exactly see it in the image. Maybe I'm just a bit blind, who knows. I think what they're trying to say is that this C has been cut out. Anyway, doesn't really matter. The idea is that we either add in a nucleotide or we take away a nucleotide. And what you could imagine is, if we were to remove this C, right, what happens is that we no longer form CAT, TCA, CAC, GTA, and so forth. We now have shifted the entire sort of what we call the reading frame of this gene. So instead of starting with CAT, we start with ATT, then continue with CAC, ACG, TAC, and so forth. And so by deleting that C at the start of this new sort of amino acid, at the start of this new sort of gene, what we've done is we've now swapped we've now changed these, right? So histidine has now formed isoleucine, right? Serine went to go form histidine, right? Histidine forms threonine, and so forth. And the idea is that we've now changed these amino acids, and it's not just changing one amino acid. Because we've now added in or have deleted an amino acid from the sequence, we've literally shifted all the triplets, and because of that, we're most likely going to be changing the resultant amino acid that's getting produced from this change, right? And you commonly see at least, a pretty big change in the resultant protein because of the effect that you have here. You're changing many, many, many triplets in the DNA. And then our final type of mutation that you guys need to know about is sort of these block mutations, which is where we're sort of cutting out a big chunk of the DNA, right? It's not just one gene that's really getting affected. We're now affecting a whole chunk of a chromosome. And so in doing this, right, there are a few different block mutations you should be aware of. Duplication, it's sort of in the name, you're just duplicating a sequence or a segment of that chromosome. So now you've got an additional amount of genes on the one chromosome. An inversion is where you switch the sort of the placement of the genes and the regulatory sequences in the same chromosome. A deletion, again, it's sort of in the name, it's where you've just completely removed a chunk of the chromosome, so you're deleting some genes. An insertion, that's where you get one chromosome, you get some of the segment of the chromosome and shove it into another chromosome, right, which is not, homologous. So this might be like chromosome 1 and this might be chromosome, let's say chromosome 20. And then a translocation is where you sort of exchange pieces of the chromosome between two non-homologous chromosomes. So again, this might be like chromosome 1 and this might be chromosome 16, for example. So I want you guys to have a look, before we go into our first break, which I'll start in the next 4 minutes, what I want you guys to do is... Actually, no, I will go and break at 2 o'clock. What I want you guys to do is I want you guys to have a look at this image over here and have a think about what has happened here, right? What type of mutation are we seeing in this bottom image, right? And yeah, that's pretty much what I want you to think about. So I'm going to give you 20 seconds to do that, and then we'll continue. Alrighty, so hopefully that's been enough time. So when looking at this, hopefully you've realised that we're swapping or exchanging pieces of chromosome from 2 different chromosomes, right? So what we've done here is we've got a bit of chromosome 9, a bit of chromosome 22, and we've done a bit of a switcheroo, right? We've swapped it around where they really shouldn't be. Now, this is a real-life example of a translocation. And what happens here is that by swapping these pieces of DNA together, right, not that you need to know this for VCE, but it's a bit interesting. What happens here is that you get this gene and you get this gene, and by swapping the chunks of chromosome around, they're now placed together. And that creates a sort of really interesting problem whereby a particular regulatory sequence here, I think it's the BCR, and then the protein, the ABL, it increases the rate of transcription, so you produce too much of this protein. And that becomes quite problematic for the cell because this is one of the main sort of causes of a particular type of cancer. From memory, I think it causes a particular type of lymphoma. And as you guys remember from immunology, this would cause the unregulated cell division of the cells, right? So it's that dysregulation of apoptosis and the dysregulation of cell division. Awesome. Just thought I'd chuck that in there. But now we're going to have a look at sort of natural selection in the next sort of 10-ish minutes. We'll try to get through as much as we can. So natural selection, I really like this meme. It's probably my favorite one that I've got in these lecture slides because it really sort of sums up the whole idea of what you guys really need to know about natural selection. So the idea here, I'll go on to the next slide in the meantime. The idea here is that when we consider populations, right, nearly every single population has some sort of variation, right? In terms of humans, it's pretty obvious, right? Some of us have, black brown hair, some of us have blonde hair, some of us have brown eyes, some of us have blue eyes, all right? So we are all different, right? Unless you have an identical twin, right? We all have some form of variation. Now that creates a sort of interesting sort of situation because despite the fact that we all are different, by our genetic makeup, right? We all sort of, if we're considering a population living in the same environment, right? These different individuals share the same environment. And what you then expect is that in this environment, there are going to be certain factors that sort of advantage some individuals which have particular, phenotypic traits over others. We can see this in the image over here, right? This is a classic textbook example of natural selection. So the hypothesis goes, or the theory goes, that if we talk about giraffes, you can, back in, ancient days, right, there were giraffes, right, and variation existed in this sort of ancestral sort of giraffe population. This variation meant that there are some giraffes which have small necks, and there are other giraffes which have longer necks. And as we know, giraffes, I'm pretty sure they're all herbivores, and they eat off the leaves of trees. Now, trees can get tall. So, what does this mean for the giraffes? It means that these little poor guys at the bottom here, right, they're a bit, their necks are a bit small, and as a result, they can't eat the leaves of the tree. What's going to happen? Unfortunately, these giraffes aren't going to survive, right? There's this selective disadvantage for these giraffes because they just simply don't have the genetic trait, to make them suitable to their environment, right? They don't have a long neck, and so they can't reach and access a food source. So they'll die of starvation. What does this mean for the big giraffe? Well, I guess it's happy days for him or her because this long-necked giraffe can then access the food source, survive, and then reproduce. And that's what this sort of next idea is. So this giraffe, which contains the alleles or, the genetic variants which give rise to the long neck, means that because this giraffe is going to survive and then reproduce, or be more likely to reproduce because they're living longer, they're going to have offspring, they're going to have children that are going to have these alleles, right? Or are more likely going to have these alleles. And so the next generation of giraffes, right, all the children or, child giraffes, right, I'm not sure if they've got a particular term, but all the little giraffes, right, they're going to have long necks too, and they're going to grow up to have long necks. And it means that generation after generation after generation, because of this environmental factor of having long trees in the environment, right, it means that the giraffe population is going to have longer necks. And this is the idea of natural selection. It's this idea that we have what we call a selection pressure. Right? And this selection pressure sort of determines which type of organisms are going to survive and which type of organisms will not. Right? And then those which are going to survive, as it says here, will then be more likely to pass on those alleles to the next generation. And so they're going to increase, in frequency, right? There are going to be more organisms in that population that have these alleles. Cool. Given that, I want you guys to have a crack at this question here. Right, I'm going to read it with you. And I'm going to give you like 2 minutes, 3 minutes. If you have your computer out, you can do this on have a go on a Word doc. I really want you to actually try answer this. Or if you've got a pen and paper, that's probably better. But I'm going to read it with you, and then I'm going to give you about 2 minutes to have a go at try to sort of answer this. And then I'm going to show you my response, or I think it's the VCAR response to this question, and we'll see how we go with it. Right, so it's a classic natural selection question, and then I'm going to go through exactly what VCAR expects when you get a natural selection question in your exam, if you do. So, this question. The blue mussel, I'm not going to pronounce these or try to pronounce them because I'll butcher the names, but the blue mussel lives along the northeastern coastline of the USA. A species of Asian shore crab was accidentally introduced into the area about 15 years ago. As shown below, the Asian shore crab has only migrated to the southern half of the total area inhabited by the blue mussel. The Asian shore crab feeds off the blue mussels. The thinner the mussel shell, the easier it is for the crab to crush and eat the mussel. In recent times, scientists have observed that the overall population of the southern blue mussel has a thicker shell than that of the northern blue mussel. This contrasts with 15 years earlier, when there was no difference in the range of shell thickness in northern and southern blue mussel populations. The question is, explain the process of natural selection that has occurred in the population of southern blue mussels over time, over the last 15 years, that has resulted in thicker shells. So I'm going to give you about 2 to 3 minutes. This is about like a 3-mark question, so expect to write, a few sentences. And then, you write that, and then I'm going to show you sort of the model response, and then you can compare. So I'll give you some time to do that. I'll give you about 1 more minute, and then at 1:57 I'll go through the answers, and then in the last 3 minutes I'll go through some of your questions that you may have popped up, and then we'll take a break at about 2:05, and then we'll resume at about 2:15. I'll give you about 15 more seconds. So just tidy up your responses, start thinking about the ideas that you put into this, or what types of points you'd put in here. Alright, 5, 4, 3, 2, 1. Alright, hopefully you guys are all good with that. So this is what VCAR says. So the 3 main points that they recommend you have in this question is that you first off have to say, right, it might sound a bit obvious because they're sort of mentioned in the question, but you have to always write, whenever you're doing a natural selection question, you have to always write that there has been variation, or that variation exists in the population. Right, so in this situation here, they say that there was variation. And I would like to be a bit more specific than that. I would say there's variation whereby some mussels have thick shells and some mussels have thin shells. Then the next point is sort of explaining what happens in terms of natural selection. So having a sentence sort of saying something along the lines of, in this environment, the crabs, the crabs which are a selection pressure will eat and kill the thin-shelled mussels, but will not, affect the thicker-shelled mussels. That's sort of your second mark. And then the third mark is then explaining the effect of that, right? So this is pretty much word for word what you'd say in the exam, right? Something along the lines of, as a result or consequently, the thicker shelled mussels survive, reproduce, and pass on their alleles to the next generation. That phrase, survive, reproduce, and pass on their alleles to the next generation, 100% just copy it word for word. It's one of those trigger phrases that you just chuck in because you use it all the time, right? Whenever you answer a question like this, which are quite frequent in the exams. Now, this is like the way I sort of answered this, or the way I would sort of direct you to go about this, right? I personally am someone who doesn't write in dot points. I write in paragraphs, but if dot points work for you, these are the 3 main ideas that you need to get across, right? The variation existed in terms of shell thickness. As I said, the crab is a selection pressure and it eats the thinner shells, the thinner shelled mussels, so that the mussels are crabs. Yeah, mussels, the thick-shelled mussels are more likely to survive and reproduce, and then that way that allele for the thick-shelled mussel will increase, so that over time I would still use that, it survives, reproduces, and then passes on that allele to the next generation, which is what that's saying there, that it gets transferred into the next generation. The final thing I want to touch on here is just this point that VCAR raises. It's a really good point because it's just such a common mistake that people make. right? And I actually will give you a few examples of this in real life. People sort of think, because of natural selection, some people have this misconception that it's the introduction of the selection pressure which drives these organisms to actually undergo these mutations and undergo these changes. So some people think, right, and as I say, it's not going to make sense because it's just not true, right? Some people think that, if I add the crabs in this environment, the mussels will just sporadically mutate to become more thicker shelled, and then they grow to become more thick shelled. That's wrong, right? That just doesn't make sense. What has happened here is that variation just exists, right? It has always existed. There's no other factor which is causing the mutations to occur. Like, these mutations happen all the time. They're completely random, and they affect anywhere in the DNA, right? They're not centered at particular places or whatnot. They're just completely random. And so because of this randomness of mutations, there are just naturally going to be some organisms which might have a particular trait and some which have another trait, right? But it's this idea that, it's not the selection pressure which is causing this new allele to come up. What it is that the selection pressure is just simply, increasing the frequency of this allele that already existed because of the variation, and decreases the frequency of the alleles that that it's killing off, or that's disadvantaged by the selection pressure. I've got some examples of this, and two really key examples that are affecting modern day, society are sort of mosquito resistance and antibiotic resistance with bacteria. So in terms of insecticide resistance with mosquitoes, what happens here is that, because of poor application of insecticides, particularly in areas where malaria is endemic or is, in very large sort of concentrations, right? What has happened is that some of the mosquitoes, because the variation already existed, right, the mosquitoes are resistant to, or have, the mosquitoes which were already resistant to the insecticide, right, survived, reproduced, and passed on their alleles, so that the mosquito populations, particularly in these red areas, are no longer responsive to the application of an insecticide. This means that governments can't really use insecticides anymore to try control malaria, which I think is, I could be wrong in saying this, but I think it's one of the biggest killers of children under 5 years old, right? Particularly in the developing world. So that's a pretty big issue that we have, right? And another sort of big example, not just with gonorrhea, but other types of bacteria too, is the development of antibiotic-resistant bacteria. And so what happens here is that because of the misapplication of antibiotics, you might be aware of, how doctors and in medicine, it's really encouraged that, doctors don't just willy-nilly give you antibiotics straight away, because, if you've got a viral infection and not just, a bacterial cold or something like that, if you've got a virus, the antibiotic isn't going to do anything, right? It's a waste. And what happens is when you misuse antibiotics, or in any medicine for that matter, you then add this selection pressure, right? Where the antibiotic is this selection pressure. And, through natural selection, what's going to happen is that you're going to have these, in this case, you're going to have gonorrhea, this bacteria, which has some form of variation. Even though they're bacteria, they still have some variation, which I won't explain how that comes about, but they do have variation in their populations. And what it means is that if a person then, uses antibiotics willy-nilly, right, what's going to happen to the gonorrhea populations is that the gonorrhea, or the bacteria which is susceptible to the antibiotic, will die off, right? Of course. But the gonorrhea which is resistant is going to survive, reproduce, and then, pass on, these alleles, these resistance genes, into the subsequent generations. So this person will effectively, have this antibiotic-resistant gonorrhea that can't be treated, and then they're going to start, you'd hope that they take caution, but if not, they will pass it on to some of their, their contacts, if I want to be a bit PG, and then they're going to pass it on and pass it on and pass it on, and that's how we see the rise of these antibiotic-resistant, bacteria, right? Because we don't really have the time for it, I won't show you the video, although it's a really fascinating video. I really recommend that you go to that website, look at the video. It just gives you a bit of an idea about the problems we face. And not that you need to know this as a specific example for, your exam, but just be aware that VCAD, like the way they write a question about natural selection is sort of like what we saw in the above, right? They'll give you a situation, like a real life situation, and then make you explain, how natural selection has occurred over time. And now they could do that with mosquitoes, they could do that with, bacteria, because again, these are things which are really prevalent in modern day society. In the last about 3-4 minutes, just to see if it's working pretty much. Okay, it's working. I'm going to go through some of these questions here, right, that you guys have. Now, there are a lot, so we won't get through them all the time, all through this period, but we're going to continuously go back to these questions throughout the lecture. So I'll answer about maybe 3 to 5 in the next, 3-ish minutes. So I've got a question up here. I'll answer this first because it's got the most likes. I'm having trouble remembering the inputs and outputs of the stages of photosynthesis and cellular respiration. Any tips? Good question. The biggest tip I've got when I sort of teach this and recommend, how students learn this is don't learn this by just writing out a table and just memorizing it, right? You're never going to remember it for your exam if you do that. What I'd like to say is think about it as a logical process, right? You're not just, when we're talking about photosynthesis and cellular respiration, you're not just talking about these random processes that are just occurring, right? That, you've got this random molecule water that comes in, and then you've got, hydrogen that just gets spit out, and you get, NAD, and then you've got, ATP that also spits out as well, right? You actually got to think about it and think logically, like how do these processes connect to each other? So in terms of, I'll use photosynthesis as an example. We all know that plants use the sun to undergo photosynthesis, right? So then what you should ask in your head is, why does the plant use the sun? Hopefully that triggers you to think it needs to use that sunlight, that energy, to break apart H2O, water. So that's one input done, water. So then we've got to think, all right, so water's going to get broken apart. What is water made up of? H2O, hydrogen, and oxygen. So what happens? Hopefully then you sort of remember that, okay, oxygen is like the waste product, it just leaves as a gas, right? And then the hydrogen, what's going to happen to the hydrogen? It gets taken up by our friend NADP to form NADPH. Another thing that I like to think about here is that, okay, this is a catabolic reaction, right? We're breaking down water, this bigger molecule, into the smaller set of atoms, right? And because of that, we're going to be producing ATP, because you might remember that catabolic reactions are exergonic, right? They produce energy. And so we produce energy here in the form of ATP. And then what do you need to form ATP? You need ADP plus PI. So to sum that all up, right, we had water as an input, we had NADP as an input, we had ADP plus PI as an input, and then as our outputs, we had, oxygen as the waste gas. We had NADPH as, the collection of hydrogens. We had ATP from that breakdown of water. And that way, I've already just logically put together what are the inputs and outputs for the light-dependent stage of photosynthesis. So if you're able to tell yourself that and, sort of say it like it's a story, or say it like it's, like a logical process when you're doing questions in the exam about this, then that's what, that's the way I recommend you do it, right? That's the way I do it. When I get a question, or if I'm doing VCE questions or testing out VCE questions that we do at Shoot Smart, and I get one on, photosynthesis and cell resp, it's not like I've just drilled into my head what the inputs and outputs are. I have to tell myself that story, right? Because that's the way I've remembered it. And that's the way I think you should remember it too. So I guess it comes down to understand the topic. Don't just sort of like memorize. Hopefully that answers your question. So I'll move that over. I'll answer about 2 more. I've been averaging 60% in bio. What kind of study score should I be expecting? Right, classic question. I've been getting X amount of, percent at school, or, I'm rank X in my school cohort. What might I get? Or can I still get this mark? Classic question. What I'd say to this is that it's hard to say, right? And I know this is not the answer you're looking for, but it's hard to say because it depends on your school cohort, right? I'll give you an example, right? So if we think about schools such as, shout out to like MacRob, Melbourne High, or, who you are when we're talking about, this sort of like higher up schools, right? Those types of schools have stronger cohorts, right? So if you're getting, I've got no idea, I didn't go to one of those schools, but if you might get, I don't know, let's say 75% in those types of schools, but that's like rank 2 and what rank 2 is getting, then you're in a good position, right? Because it's, you're in a strong cohort and you're ranked well in that cohort. But if we're talking about schools which might not have a strong cohort, then you might be rank 1 getting, great marks, but if those types of questions that you're getting aren't reflective of, not reflective of what VCAR actually is, or if you are rank 1 in a cohort that isn't very strong, it might be a bit of a false sense of security, right? So for instance, I wasn't rank 1 at my school, but yet I still got the highest study score at my school. And likewise, there were people who weren't, rank 3 or rank 1. There are some people who were like rank 5, 6, 7 that still were able to get, relatively higher scores. Like I think, I could be wrong in saying this, but I think there was like a kid who got like rank 5 and then ended up getting, 40 to 45. I can't exactly remember what score it was. But the idea is you don't necessarily need to be, getting X amount of marks or, whatever it may be. The first point I want to say is that you need to focus more on the rank, because that's at the end of the day what VCAR looks at. They don't really care if you've been getting 60% or 90% or 40%, right? They look at your rank. And then on top of the rank, you need to also then consider, where do I rank in my cohort, and what type of cohort am I in? Am I in a really strong group of kids, right? That's not to say that, stereotypically poorer school, not economically poor, but stereotypically low scoring school is going to be bad, right? Sometimes some cohorts, it might be at a school which you don't think is like a higher up school, but the cohort is actually a strong cohort, right? There are some schools that do really well in particular subjects, and so what I'd say is, it depends on how strong your cohort is, and what type of marks you're getting in that cohort. Right, and in the sense of your rank. Again, doesn't answer your question, but what I'd say on top of all of what I've just said is don't stress too much. What I'd say is try and implement those things that I said at the start of the lecture, right? Start doing things like practice questions if you haven't already. Start trying to make your own notes and understand those notes if you haven't already. And try, even if you are doing those things, maybe start thinking about what other things can I be doing, right? Are you studying, really hard, up until I don't know if I never did this, but I don't know if anyone does, but, if you're studying up until 3 a.m., 4 a.m., pulling all-nighters to study for a SAC, maybe that's your problem, right? You might be thinking, I studied so hard, but if you're not studying right, then obviously it's not going to work out. So there's some of, my big advice. So start, if you feel like you're doing a lot of work and yet the marks aren't, showing through, I'd probably recommend start maybe thinking about other things as well, which might be impacting your marks, such as, things like sleep, exercise, other stuff like that too. Cool. So hopefully that answers your question. But at the end of the day, it really depends on your exam as well. If you smashed your exam, you can still do pretty well. And I'll just finish up with this last question here before we take a break. So how much I assume that's meant to say how much biology study? How much biology study did you do leading up to the exam, and what did you do to obtain a 46 raw? Good question. What I'd say here is, I didn't have a timetable of sorts. I didn't be I'm putting the timer on, 4 hours, tick, 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 done. I'm going to study now for 4 hours. That's not what I did. And, when you talk to people who do well, it's not like they said that, I'm doing 10 hours of bio this week and, 15 hours of this subject this week. Because, I don't know about you, but if I try and make a timetable, I don't even follow it half the time. You know what I mean? So I'm bad with timetables and organisation like that. So what did I do? I sort of just went off my gut feeling, and if I felt like I needed to learn something, I just went off and studied it, if that makes sense. So to sort of answer your question, most nights I'd say I studied, right? I'd say it's pretty uncommon or quite rare when there was a night where I just didn't do any work. There were obviously some nights where I just did nothing, or I just watched TV, or just chilled out with my family and stuff, because you need those times to just have a break. But I felt like most nights I did do work, and that's quite frankly because I just felt like, hey, Michael, I need to get ahead with this topic, or hey, Michael, I didn't quite understand this topic well, let me go over this topic again, or hey, Michael, let's do some practice questions on this particular topic. So it really just depends on what you're feeling in terms of leading up to, I guess, in terms of your study, right? If you're feeling pretty confident with stuff, do practice questions. If you don't feel confident with stuff, review your notes and try, make sure you understand it. What I'd also say is that particularly in terms of the lead up of the exam, what I did was practice exams. I did spend a few days just brushing up in Unit 3 stuff because I was a bit slack. Should have probably looked at Unit 3 stuff a bit earlier, but I didn't. But anyway, so I spent a few days after I finished Unit 4 just to brush up on Unit 3, did a few Unit 3 practice exams, started doing Unit 4 practice exams, so before 2012 exams when they used to be Unit 3 and then Unit 4 exams as opposed to just an end of year exam, which is Unit 3 and Unit 4. That's sort of what I did, and I was doing about, I'd say, I can't exactly remember to be honest, but I would say I did at least one a day, because it's not that hard, right? You can, from memory, they didn't take too long. You just have to calculate the exam time that you do to do the exam, plus maybe an hour to mark, or 30 minutes or 40 minutes to mark, and make sure that you're understanding why you're getting questions wrong. Hopefully that answers, and that's pretty much how I got a good score, right? By doing lots of practice questions and exposing myself to those questions, I made sure that I made sure that I knew what to write for my exam. Alrighty, so I'm going to leave it there, right? We'll resume again in a moment. So I'll leave you with this slide here, right? You guys can just chill out for a bit. We'll make a start again at about, let's say, 2:25, right? So we'll start back again at about 8 minutes, because I did go a bit over time, but we'll start again 8 minutes time, and then we'll continue on with the stuff. So I'll see you guys then, right? 2:25, where I recommend get up, have a stretch, walk around, get a drink of water, go to the bathroom if you need, whatever you need to do, right? And then we'll get right back into it. So I'll see you guys then. Alrighty, we'll make a start again because it looks like we've reached time, which is cool. Alright, so 2:25, we'll take the next break at about, I don't know, let's say 3:15, maybe? Yeah, not. Okay, so let me just write this down. So, yeah, about 3:15. So 3:15 to 3:10. Alrighty, so just before we make a start, I just wanted to quickly tell you guys about one of the things that we do with atarnotes, right? And you may have heard about this before, but this is called, we've got a thing called Ed Unlimited, right? So I'll quickly show you guys what that is all about. So give me 1 second, right? Let me just quickly find it. There we go. Okay, so Ed Unlimited, what is it, right? So Ed Unlimited is effectively a sort of a thing that that atarnotes does, right? And it's pretty much, the way we like to sort of describe it is it's like the Netflix for nerds, right? It's pretty much like Netflix in that you've got, access to all, a whole bunch of different TV shows and movies. If you if you're keen, The Sinner is pretty good. I just binge watched like season 3 in the past few days. But in terms of Ed Unlimited, what we've got here is pretty much access to a whole range of our publications, right? And this is publications for, it would be all your subjects, right? As well as further publications from other companies such as NEAT. Now, what does this actually mean? So just going through, as you can see, the list is it's a pretty long list, but just to show you what I mean by this, right, if my cursor works, there we go, right, so say these are the subjects that we're doing, right, and look at this, Bio 3-4 is right here, we all love Bio 3-4, so let's have a look, right, so what you get with this is, for all your subjects, right, if we, offer the publications, of course, right, you would get access to things like our course notes. So in terms of Bio 3-4, we can see, let's say we go to Unit 4, and I want to have a look at, I don't know, let's say the causes of variation, right? There you go, pull that up, right? And here's just an additional, sort of resource that you guys have access to that gives you a bit of an idea or further sort of understanding about the course, which is something that you can also look at with your textbook, right? So you might be looking at your textbook and thinking, do I really need to know this? Or do I, have I learned everything that I need to know, especially when you're looking at the study design? And so, looking at our course notes, that gives you a bit of a rough idea about, okay, this makes sense, and this is relevant. Or it might also give you the ability to sort of see like, I didn't actually consider this topic in this way, right? I never actually thought about it in this way, because obviously the way your textbook is written is going to be the way that this is written. It's really written in a sort of like a student perspective and a bit more of an informal manner to help you sort of better relate and sort of understand with the subject content. Now, obviously I work for atarnotes, and you might see this as being a bit biased and stuff like that, but like 100%, I'm not even like bullshitting you right now. I use the course notes for bio from atarnotes, and like I swear to God, like it was actually the best resource I used for bio, hands down. It was like I don't know, it's a bit bad that like obviously I'm wearing the atarnotes uniform and all of that sort of stuff, and you might think, Michael, what are you doing? Like you're working for atarnotes, this is obviously crap. I'm not joking you. This is one of the best resources you can use, hands down. I'm not even like 100%. If I had a little brother that was doing bio, I would get in this. If I had like a family friend that was doing it, I'd recommend them this. You know what I mean? It's honestly like one of the best resources you can use. Now, in addition to Ed Unlimited, what you can also do is there's sort of other functionalities you can do. So I know you can like highlight. I'm not meant to do this because apparently this sort of highlights the book for all the other tutors, but who cares, right? So you can highlight whatever you want. You can write things down. Or maybe you draw. I don't know. Yeah, you can draw. I think if I use the pen, there you go. I can draw. Oh, there we go. That makes it good. I can zoom in, zoom out. I could search up keywords. I can be like, I want to look at natural selection. So let's go natural selection, searching. There we go. And that will bring us to a bit. Okay, here we're talking about immunology. I should probably write natural selection. But anyway, you get the idea, right? So these are our course notes. Another thing that you can get through Ed Unlimited, as I've said, right, it's got a whole library of different sort of things that you can have a look at, is, obviously the course notes. For biology specifically, you can also get access to our topic test. It's a bit of a chunky boy, right? So I've got it right here with me, just to show you the physical version. I have to say, she's pretty thick, right? And what you can see in the topic tests is that it's a bit like, I don't know, checkpoints, if you've ever heard of checkpoints before. But what topic tests are all about, it's pretty much the different sort of topics or concepts in bio grouped together as questions. So it's really a question book, right? And this is probably my biggest recommendation if you want to try and improve on your weaknesses, right? So I mentioned that earlier on. Say immunology was your weakness, right? Immunity and immunology. Say you didn't quite understand that properly. You can go and say, all right, I want to practice some questions on this, and you can go through. And this is arranged as like a test with, full on, here, I'm pretty sure it outlines as well, like how long you should be spending on doing these questions. And that way you can sort of test yourself and practice with, questions. The great thing about these topic tests is that it's not just the VCAR questions in here, right? So it's not just like we just ripped off VCAR, put their questions in, in order of topic and then selling it to you, right? That's a bit of a scam, right? It's a mix of VCAR questions and our own questions that we've written, right? And I'd like to say that we write pretty good questions. Maybe that's just my ego, but anyway. But in addition to the Shoot Smart questions or the atarnotes questions that we've written, there are VCAR questions which are important to look at as well, to also to get you guys to have an idea about, what is the difficulty of VCAR, and then if you really want, you can go onto the exam report and then have a look at, how many people got that question right in that exam. Another thing I'd like to show you with these topic tests is that if you have a look at the solutions, particularly for the short answers, we've written out exactly how you should have written these for like an exam situation. So if it loads, I don't know why it's not loading now. Here we go. Okay, looks like we're going to do a few pages. Let's get through these multi-choice. Right, so multi-choice obviously have explanations and stuff like that. Did I skip it? Maybe I did. Now, here we go. There you go. So we sort of give you an idea about, how much you can write. So in my personal perspective, what I found with, other sort of resources are question resources that I used from different companies, right? Again, not the bias, like this is honestly my own honest opinion. What I found was that there are some books which actually did have good questions in them, like VCAR questions and stuff as well. But their solutions or their suggested solutions written by the authors of those books were just too long. You could tell that they just typed it out on Word, right? And whenever you type out a response, you end up writing way more than what you'd write on hand on paper. And as a result, what you find is that, what I found at least with those types of books was that the solutions were often way too long for what you'd actually write in the real exam, and it sort of doesn't really show you how to write concisely. Another thing I'd like to say and so like related to that point is that then you write concisely by looking at these sort of model solutions that our tutors have written, again, hand on paper. So I think that's pretty cool. What else is there? So that's bio, sort of the topic test and the sort of the course notes, but then you get other things too, right? So you get access to NEEP questions as well. So their NEEP questions book, which I can quickly show you. So let's go through. So again, like the topic test, this isn't in an exam situation or like exam format, but you can go through this to have a look at, to summarize or, to revise particular topics, right? So you can just attack a few of these questions every day or, once a week to just continuously revise. And then the NEEP book also has an exams book, which is sort of like a bit more like our topic test, which sort of arranges, it's got a variety of exams in there and it's written in sort of an exam style. So as you can see here, right, you've got, multi choice and then you've got your short answer sections and all that sort of stuff to mimic the real thing. And then, in addition to that, right, this is taking a lot longer than I wanted to, but it's because there's so much in here. In addition to this, you've also got things like NEEP exams which aren't a part of this book as well. So these are trial exams, like company exams that teachers usually buy, I'm pretty sure, and like give to students at the end of the year, right? So in case your school doesn't give you exams and that sort of thing, right? We've also got access to NEEP exam resources in an Unlimited 2 that you guys can use as an additional resource to further your studies, right? There's also things like the NEEP notes, like the NEEP biology notes, and also the atarnotes practice exams that we wrote last year. I was involved in writing one of them. Had to say, again, it might be my ego, but I'm pretty sure my questions were pretty good. But anyway, anything else I should probably let you know? I guess for the kids that are doing English, I did English language, so, it sucks to be you guys. We are better, the England students out there. So shout out to all the England people. Lit, it's a bit like, literature's meh, but then English, you guys are just plebs. Anyway, but if you guys are doing English, I'm just trying to find where they are. Where are you? Okay, there are some here. If you are doing English or I think literature as well, you guys look at books too. We don't, so we're lucky. But if you're doing English or lit, we've got what we call our new text guides, right, which are written by some of our tutors and other people who are experts in these sort of books and got really good in English or literature and have pretty much analysed them for you. And I assume that would be pretty helpful. Again, I don't do English, but I guess that's always, the more resources you have, the better. And with it Unlimited, you get a ton of resources that I think you can appreciate from just the amount I'm showing you. Awesome. So you might think, okay, Michael, what's the catch? What's the go? This must be super, super, super pricey and super expensive. Thankfully, it's not, right? Even for my standards, as a uni student that's pretty broke, right? It's not that expensive. So with today's lecture, right, a bit of I feel like I'm a YouTuber now. I said this comment last lecture series, but if you use my promo code MichaelBVCE rhymes, you can get pretty much a month, right, free 28-day trial of the Ed Unlimited website. So there's nothing to lose, right? If you think I'm just a bit of a sellout, that's okay. Have a go, right? See it for yourself. If you like it, you like it. If you don't, you don't, right? It's your opinion. You don't have to trust my word for it, even though I think it's great. And if I was in VCE, I would do it. But you don't have to trust my opinion. You can use the promo code, go, make an account, use it for 28 days. If you like it, you can continue. I'm pretty sure it's pretty cheap. It's what you'd be paying for your Spotify or your Netflix monthly sort of payments anyway after that 28 days. So it's nothing expensive. And if you don't like it, in that free trial, then we've just given you a whole bunch of our resources for free for a whole month. Right? So it's a bit of a no-brainer. If I was a student, I'd be hopping onto this right away. Again, I feel like I actually don't have to explain it to you guys. It's a bit silly if you don't, but it's a really, really generous offer from the atarnotes, from atarnotes. So again, if you use this sneaky promo code, right, then you guys are set. So I've got you guys, right? And I just, had to put a meme with it because I love my memes, right? It's honestly such a good deal, right? So if you look on your website, I'm pretty sure, right, and you scroll down to where you like write your questions, I think on the right hand side they've got some links to Ed Unlimited and Shoot Smart, right? If you go to the Ed Unlimited link, you don't have to, obviously, you know, you don't have to do it like right now because we're going to continue with the lecture content really soon. But just make sure that you keep a note of that tag, right? And that way you can get that free for 28-day trial. Again, I just want to finally, the last time I'm going to say it, if you use that, you know, that code, Michael VCE, right, there's nothing to lose. You literally have nothing to lose because you can try it out. If you don't like it, you just stop, right? You opt out. If you do enjoy it, then fantastic, right? You continue on. But we've effectively given you all our atarnotes resources for free for 28 days. So I don't think I have to explain that any further. Just test it out for yourselves. Now, another thing we've got going on is if you do want, you know, a physical copy, right? If you do want to like hold this, you know, this thick boy right here, right, in your hands, and you don't want it on a computer, that's cool too, right? And so we've got an offer for those of you who do want the physical copy, and in that case, you just use the promo code July15, and you can get 15% off any of our sort of books that you can buy online on the atarnotes website. But that said though, why just buy the physical book when you can save so much more money and get access to all our books for, you know, every month? It's a no-brainer. Anyway, that's my opinion. Anyway, let's go back to the content. So, going back, where were we? We were up to selective breeding. There we go. Alright, so continuing on, selective breeding. So this is slightly, it's related to natural selection, but it's slightly different because what's happening in selective breeding? In selective breeding, well, the wild and the nature in the environment is not the selection pressure. In selective breeding, the humans or the breeders in this situation are acting as the selection pressure, right? So it's not really a natural circumstance anymore. What's happened here is that, you know, as humans, as breeders, we've said, okay, I don't know, let's say I want cows that produce a lot of milk because as a farmer, I want to make the most amount of milk to sell to Coles and Woolies, and then I want to make the most amount of money. So how can, as a farmer, make the most amount of money? The way I can do this is by getting my best cows that produce the most amount of milk and pair them up with, I think the male version of the cow is a bull, maybe, I don't know. Anyway, you pair them up, right, and then they're going to pump out kids that are going to, you know, they're going to pump out cows which are going to produce a lot of milk, right, and that means more milk to sell to the Coles and Woolies, right, and that means I'm a happy farmer because I've got more yield of produce and therefore more money in my bank account, right, we all love that. What else did I have to say? Yeah, and in terms of selective breeding, it also means that, you know, if I've got a cow that's not producing lots of milk, right, we're going to forget about her, right, because it's useless. They're not making me money, right? As a farmer, I want to make the money. So we're going to, you know, ship her off to a different area of the farm. She's not going to reproduce and have kids that don't produce milk. So through selective breeding, you know, jokes aside, through selective breeding, we produce, the idea is that the breeder, based on what traits they deem to be desirable, right, they then select the organisms which express those or have those traits, put them together, reproduce them together, and then we have generation after generation, we produce this sort of this population of organisms which have this desirable trait. And so the idea is obviously to benefit the breeder. I've given you the example of the farmer and cows. So you want cows to produce milk, and therefore, you know, the more milk production you get, the more money you're going to make. You can apply that same example to, I don't know, let's say dogs, right? So I used to have a pug, right? What would have happened ages ago is that people would have thought, you know, ancient days, they would have thought, oh yeah, let's look at this dog, right? I want to make a dog and have it, you know, flat face, right? And they would have picked, you know, the dogs with the shortest muzzles, bred them together, and over time would have developed what we know as a pug, right? Pugs are awesome, I love pugs. But the thing is, right, it's not really good for the organism, right, in some cases. And the reason is because we sort of decrease the genetic variation in these populations because of what we're doing, right? We're saying that we are only going to have these, these particular genetic traits and only put them together, right? And we're going to make sure that no other organism, right, in the population that doesn't have these genetic traits, these alleles, they are not going to reproduce. And that might not be great for the organism because it might lead to health effects, right, or disadvantageous health impacts, right? So I'm no like vegan or like animal or plant animal activist. Yeah, that's it. Animal activist, right? I'm a woke, right? But I do live in Brunswick, so I sort of have the vibe of the greenie life. I must admit, it's pretty nice having a keep cup and going to the cafe and getting my avo and toast, right? We all love that. But in terms of chickens, right, I'm going to give you an example of chickens as a disadvantage of selective breeding. So what happens here is that the chickens, right, over time, the farmers thought, okay, I want chickens to be bigger, right, because obviously the bigger the chicken, right, the more muscly the chicken, the more chicken meat I can sell off to Coles and Woolies and the butcher, and the more money I can make. Right, and I assume the bigger the chicken, the bigger the eggs, and we all love eggs, right? I love my poached eggs and my avo and toast, so I'm going to pay money to have these eggs. So, what has the farmer done? They bred the biggest chickens with the biggest chickens to then produce children, which are going to be big chickens, right? And over time, that's led to this increase in average mass or average weight of the chickens. So, enough about chickens, right? What's the deal, Michael? Right, get to the point. The idea here is that what has happened with these chickens is that they're actually so large that they can't really support themselves on their two legs because their two legs can't really support the amount of weight that has been sort of put at the front of their body, right? And so what happens is these chickens really struggle to walk, right, and fall over a lot. And you could imagine then if you put these chickens in the natural environment, right, not in a farm cultivated environment, but like in a natural environment, what's going to happen is that these chickens aren't going to live, right? They're not going to support themselves or survive because if they can barely walk and they struggle to walk, right, they're going to, die in the nature, right? They might get eaten up by a fox or something, I don't know. So that's to give you an example that, selective breeding, although it's great for the farmer, usually on an economic basis, it doesn't always lead to the best health impacts on the organism. Alrighty, so to continue on with this idea of variation, genetic variation and stuff, right, and evolution, we have to talk about gene flow, and the VCAR definition of gene flow, which I've got right here, is that it's the movement of alleles between populations, right, and they say by interbreeding. So it's the movement of alleles between two populations. So what we have here is one population of a particular bird, another population of a particular bird. They are the same species. Right, that's what a population means. Like when we're talking about populations, right, in this situation we're talking about populations of the same species. They're just in two different geographical locations. So what's the idea here? The idea is that you've got this bird which has like the dark blue, right, which is represented by little a. They are emigrating, so they're leaving this population on the left-hand side, and they are immigrating into, or they're not colonizing. They're immigrating to this population over here, right? And as you can see, in doing this, right, they're introducing these alleles, right, little a, into this new population. So it's actually increasing the genetic diversity of the sort of where the bird is going. What does it mean for the population which is having its members leave? So what does it mean for emigration to occur? If emigration occurs, because individuals will be leaving, right, we might see a reduction in genetic diversity because alleles are getting taken out of the population. Awesome. Will we do this question? Because of time, I might skip it. Yeah, because of time, I might just do the multi-choice that I've got in here. So I'm going to skip this question, but after this lecture, you guys have the slides. You've got this question here. It is a VCAR question, so you can look at the VCAR answer. Otherwise, you've also got the recording, so you can just watch this back, You can see this slide, which I've just left up for 5-10 seconds, and you can have a look at the answers. But it's from the 2017 exam, so you can just have a look at the 2017 exam report to check your response. All right, so the genetic drift, what does this mean? Right, another way we can get sort of a change in genetics is through a change in allele frequencies, right, or the change in the proportion of the alleles, right, in populations. And what happens here is that these are changes in the amount of alleles, or the proportion of these alleles, and they're changes to them as a result of a chance event, or as a result of just random fluctuations, right? And what does that actually mean by this chance event? We'll have a look at this in terms of the bottleneck effect and the founder effect. So in terms of the bottleneck effect, what happens here is that a population gets subjected to what Vika has actually mentioned in a definition question, becomes subjected to a catastrophic event, right? So that might be, they get subjected to, bushfires, right? This is what we would have seen in, Australian outback in January, right, and December. So, if we use that example, right, the bushfires would have wiped out large members of the communities in those areas, right, the animals and plants in those areas, and obviously that's by chance, right? And so this leads to this massive reduction in the population size, so that the new population becomes unrepresentative of the old population. What does that actually mean? If you look on the left-hand side here, we've got 3 red, we've got 3 green, and we've got 3 blue. That's our sort of frequency, or our proportions, right? We've got 3 red to 3 green to 3 blue. If I want to summarize that, I could just say 1 red to 1 green to 1 blue. Now, we've got a catastrophic event, right? In this case, it's a fire, right? Let's say a bushfire. The new population, the proportion is 3 red to 1 green. You can see that frequency is very different to the old frequency, right? One of the biggest things we see is that blue has been completely killed off, right? We don't have any more of this blue allele anymore. This means that if we try to recover these endangered species, or these species that have just come through a bottleneck effect, you'll never, or it's very unlikely that you'll ever reach the same genetic diversity you had prior to this catastrophic event. So what you see here is after the population sort of grows again, we don't have any blue alleles anymore, because obviously we've wiped all those alleles. The only way you could ever get that again is if you get, immigration or gene flow from another population, or if you perhaps get a mutation that leads to that, the production of that allele, right? But that's very unlikely. And so it's very hard to reach that same genetic diversity again. Another thing you notice is that because the 3 red, or I guess red is now a much greater proportion than the green, you're going to see that again in the new population, right? Where red is going to be more common than the green sort of allele, which is shown here. And then ultimately, this leads to less genetic diversity. Now, the founder effect, this is pretty much a process in which you get pioneers. So you get these a small group of members from a population that just randomly venture out and colonize a new area, a new geographical area, and establish a population there. Right? And it's been hypothesized that by doing this, through the founder effect, it sort of explains why there are particular sort of, let's say, diseases or conditions which are at higher frequency in small islands that may have come about because of the founder effect, compared to other sort of areas, geographical areas. And one example of that is in Tasmania, right? What you see with Tasmania is that there's a particular condition called Huntington's disease. It's a neurological disorder that leads to, I think over time, it leads to the breakdown or the degeneration of the brain. It's in a much higher frequency or proportion in Tasmania as opposed to other areas of the world. So, insert, Tasmania joke, right? And insert joke about Tasmanians being a bit, slower than everyone else. But no, they do have, there is a higher frequency of Huntington's disease in Tasmania, and it's because of, it's hypothesized to be because of this founder effect. And just to reiterate that this is more common when you get this small population size, because when you have small population sizes, it's much more random, or it's much more volatile about what's going to happen in the next generation. And you sort of see that in this image here. It's not that, it's not like the blue balls are better at surviving in the jar, right? Assuming these are like organisms, than the red ones, right? It's just, randomly by chance, the blue ones are just reproducing and replicating more often than the red ones. Alrighty. The next concept we're going to go through is what we call allopatric speciation. So what happens here is that, I'll show you the image, I think that makes it the most sense. So what happens here is you start off with this original population, right? So you have a population of particularly like an ancestral species, so of like a species, and what happens is you get this like division of the population, right? A barrier of sorts. This barrier might be things like a lake or a mountain range that develops over years, or a flood which causes, which forms a lake in between the two populations. And the idea here is that this geographical barrier now prevents gene flow from the two, separated populations. So the members in the left-hand side can no longer immigrate and emigrate, in the population on the right-hand side. And this means that we no longer get reproduction between these two populations. This means that over years and over time, generation after generation, you'd expect that potentially the population on the right might be subjected to, selection pressures which aren't found in the population on the left-hand side, or it might be that there are differences in mutations that pop up. So organisms on the left-hand side accumulate different mutations that are than the ones on the right-hand side because of just random chance. And what this means is that over generation after generation, these organisms are going to become really, really, really different, and they become so different that if you then were to bring them back together, and if you attempted to reproduce them together, right, if you attempted to put them together and make them mate with each other, they wouldn't be able to produce a fertile and viable offspring. This is the test we use to determine if one species is different from another. So I'll give you another example of this, right? If you were to put a dog with a cat, right, and you wanted to try and produce a hybrid between a dog and a cat, it's not going to happen, right? We all know that's not going to happen, right? Even if you managed to get a dog to reproduce and mate with a cat, right, which is just weird, but even if you managed to do that, right, the female wouldn't get pregnant, right? And it's because the organisms are so different, right? They're so different genetically and in other forms and other ways that they can no longer produce a fertile and viable offspring. And this is the test we use to say that one organism is a different species than another organism. You might be wondering then, hey Michael, I remember hearing, on a David Attenborough doco that lions have mated with, or like let's say zebras have mated with a donkey, right? And they've mated a zonkey. I think that's what they call them, right? The mix, right? A zebra and a donkey forms a zonkey or some shit like that, right? So you might be wondering, okay, how does that work? A donkey and a zebra is different, right? They're different species. How do they make an offspring? Again, I'm just going to circle this one more time. The offspring needs to be viable and fertile. In that case, right, the organism is viable. That means that the organism doesn't die during pregnancy, right, or that pregnancy can actually work. So a non-viable offspring would be one that, died through miscarriage or just never really formed an embryo properly or whatever it may be, right? It never gave birth to the offspring. And it needs to be fertile, meaning that that offspring produced needs to be able to actually have children. The thing with the zonkey and these other sort of like hybrid animals or other sort of organisms is that they are infertile. Right? They can never have children of their own or offspring of their own, and that's why a donkey and a zebra isn't considered what was I going to say? Isn't considered different species, or they aren't considered different species. So if you're ever answering a question on allopatric speciation, just be aware of this mark allocation. Like the natural selection questions, these are quite stereotypical, they pop up quite often, and when they do pop up, they usually pop up in the same sort of way, right? And this is the general way that you go about answering them. So the first sort of mark you usually get is by saying that there's, geographical isolation separating the population, and that prevents gene flow. The second mark is by saying something along the lines that, that because they have now separated and they're sort of living in their own environments, right, they'll be subjected to different selection pressures, and I like to say that they'll accumulate different mutations over time. This means that over the years, over time, or over a long period of time, right, there are going to be so many, there's going to be such a large extent of difference that if these populations are brought back together, right, they will be unable to produce fertile and viable offspring. So that's generally your mark layout. I am going to do a practice run of a question, so I'm going to give you 2 minutes or 3 minutes to do this. I've sort of just gone through how to answer this, but just to get you to practice and to make sure you remember, because it is important. If it does pop up on your exam, you can thank me later. But here's sort of a question that we've written at Tutsma. So originally there was one large population of gazelles living in Africa, but then during the wet season, extreme flooding occurred. This flooding formed a large river that the gazelles could not cross, and which separated half of the original gazelle population from the other. If this river then persisted even during the dry season, explain how it could lead to the formation of a new species. Have a go at that. I'm going to give you, I'll give you 2 minutes starting from now, right, and then I'll go through the solutions. Give you 30 more seconds. Alright, so I might go through the solutions now. So this is sort of like, I can't remember if I wrote this or one of the other tutors wrote it at Tutsma, but this is sort of what we came up with. So the river could lead to the formation of a new species via allopatric speciation. The river would act as a geographical barrier preventing gene flow, 1 mark, between the two populations of gazelles. The gazelles on the opposite sides of the river may have different mutations arise in the groups and/or would experience different selection pressures from each other. So that's like your second mark.
And then this means that over time, allele frequencies would change in the different groups, and they would develop different traits. Eventually, the two groups would become so different from each other that if they were once again brought together, they would not be able to produce viable, fertile offspring. So make sure that you note that down, because that is a pretty important concept to understand. Alrighty, so what we're going to do now, so I just got up my computer to activate the polls for these. So what we're going to do now is we're going to have a go at doing some of these questions, right? And this is going to be interactive, so it won't just be like me sitting here waiting for you guys to do the short answer questions. We'll actually be doing these together and do a poll, and I can show you exactly, you know, what your peers have put in the and show you like the percentage of people getting it right and all that sort of stuff, which I think is pretty cool. So looking at this, right, looking at this question, so consider the diagram below that models changes in allele frequencies for one trait in a population over 2 generations. The original population is shown on the left. If the diagram above models the founder effect, then event X is what? So have a go. I'm going to activate this poll, right, and you can put whatever answer you feel is the correct answer. So I'll give you maybe 20 more seconds, let you have a think about it. Right, 5, 4, 3, 2, 1. Alright, so I've just locked the voting. You should be able to change your votes now. And we've got 200 votes, so that's pretty good. Right, and 70% of you put A. Now, I'm really not sure if you guys can actually see what each of you have actually put down. You guys got it right. I just realised while just checking this that I accidentally miswrote the question. I said B, but it's actually A. If you put A, you're correct. B is if it's the bottleneck effect, and I think I just misread the question when I was doing this. So there you go, goes up to A, that's good, right? Let me just have a see what you guys can actually look, like if I go present mode, right? So you guys see this, I think, I don't know. Hopefully you guys aren't seeing what everyone else is putting, so then everyone just puts on A, right? Hopefully that's not what's happening, but nonetheless, it's A. Let's do the next one. So let me stop this. Alright, and I'll do it again. I'll read it out first. A patient with HIV subtype O was treated with drug QT. After a few weeks of treatment, all of the HIV in the patient was resistant to drug QT. This drug is most likely due to what? Okay, that's not what I want to do. Let me try restart. Reset results. Reset. Done. Cool. What do you think about the HIV question? Okay, I just unlocked the voting. I just realised I had the voting locked. Sorry. Interesting. Okay. I've got quite a large number. We're approaching 300 people. I just want to say, don't be affected by the peer pressure. If you're, I don't know what you guys see on your screen, but if you're seeing what I'm seeing, which is 80% of people saying, oh, actually, no, it's all good, it's all good. No, my bad. I thought the poll was in order. It's all good. Alright, I'll lock the results now. Okay. So you guys are right. On my screen, I was like, why is 80% of people saying A? Like, we just went through natural selection for like 20 minutes. I would be so like upset. Okay, that's good. All right, 80% of you, so 79% of you put C, which is the right answer. Fantastic, right? This goes back to the idea I was telling you about, that natural selection doesn't mean that, you know, in this situation that HIV doesn't just see the drug and is like, oh, yep, I'm going to mutate, right? It's the fact that there are already a variety of the viruses, right? There's diversity in the population, so that when you put in one drug, right, there's, you know, pretty good chance that one of the HIV, you know, subpopulation or members in the population are going to be resistant, right? And they're going to then relapse and form this relapsing infection. Which really, I mean, I'm going off topic here, but it's really the biggest sort of struggle that happened with HIV back in, you know, the 80s and 90s when HIV sort of, you know, came to light as a pretty big thing in, you know, I guess like the Western world. And a lot of people really died because of this, because, you know, when the first drug was sort of like discovered to treat HIV, right, what people didn't realize, because our knowledge of medicine back then wasn't the same as it is now, was that, you know, people were being prescribed one drug like only until when they were like super sick, right? But they were only being prescribed one drug, right? And so what happened there was HIV was just becoming resistant, right? And it just got to this sort of cat and mice game where, you know, the patient would be given a drug, they'd be better for like a few weeks and then, or like a week or two, and then there would be a relapse of the infection, right? Because the HIV would re-emerge every single time, right? That's got to do with the nature of HIV and how HIV is such a complicated virus. It's super fascinating. I think it's so interesting. Anyway, I'm rambling. Let me continue. Otherwise, we're never going to finish. So, unlock the voting. Cool. All right, let's look at this next question. Over the past million years, Australia's climate has become much drier, leading to reduced areas of forest and woodland. Studies of mitochondrial DNA in different spider species from various woodlands show they share a recent common ancestor dating back to just before the climate began to become drier. The different species are likely to have evolved through the process known as what? Let me start the poll. Oh. We're going to reset the results, my bad. Alright, let's play. Cool. When you're ready, just chuck in your answers. Pretty good signs. You guys are a pretty smart bunch, which is good. Alright, 5. I'll wait till like 100 or so more of you vote. So at the moment we're approaching 200 people voting, which is pretty cool. 10, 9, 8, put in your vote. 7, 6, 5, 4, 3, 2, 1. Alright, we've got about 300 people. That's pretty good. Alright, so I'm going to lock it now. Cool. 74% said C. Right, and you guys are right. Fantastic. So that's pretty good. Now, is this the last one? I don't know. I think this might be the last one before we go onto new content. So I'll let you guys do this. Let me just reset the results and we'll continue on. So we'll reset the results, reset, and then we're going to play, unlock. Cool. So in populations, answer whichever one you think is the correct answer. I'm only going to give you like a minute to do this because we're running out of time. And then I might, depending on what the content is next, I might go through another set of questions you guys have. We'll take then a 10-minute break, and then we'll continue on with the rest of the lecture. Okay, this one's good. We've got a bit more of a split happening, maybe. Okay, so 10, 9, put in your vote. 7, 8, 7, I can count. 6, 5, 4, 3, 2, 1. I'm looking at now. There we go. Cool. Alrighty. So about 75% of you said A, and you guys are right. Fantastic stuff. Awesome. So here's just a short answer that I thought would be good to do, but obviously we're running it out of time, so I'm just going to skip it for now. What was I going to say? Yeah, so have a look at this. You guys, as I said, you've got the resources, you've got the slides, so you can have a look at this in your own time. This is from the sample exam, and this is my suggested solution, right? So again, you've also got this recording, so you can just rewind it back to this point. I'm going to leave it here for like 5 seconds. You guys can, stop it, pause it when you want to check the answer. Again, this is a suggested solution, right, that I wrote. Who knows, like if VCA would write something differently or whatever it might be. I think from memory when I wrote this, there was another sort of answer that I thought you could probably write. So don't stress if your answer's not exactly what I've written here. But yeah, again, there are no VCA solutions for the sample exam. So it's a bit hard to think exactly what they want you to write, but this is sort of what I'd say. Awesome. Okay, I think this is the last one, and I really like this question. So I'm just going to do this one, and then we'll go through some questions. So reset, play this, and unlock. So the wild sunflower plant has been cultivated by humans over several generations. During that time, selection for or against particular sunflower traits has been carried out. A comparison of some of the traits in wild and cultivated sunflowers is given below. From the information above, we can assume that humans have selected against large what? We've got a direct 50-50 split at the moment. This is honestly a question that you don't even have to do biology to get this, right? I could give this to like my mum or my dad, and maybe, I don't know, maybe you'd hope that they'd be able to look at this, interpret a graph or results and be like, yep, right, this is the answer. You don't need to actually know biology to get this answer. Alright, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Alright, we've got about 200 people, that's good. Okay, about 66% of you said B, and you guys are right. Honestly, the only thing you need to see is which one has which one's the odd one out, right? If you had no idea, right, that's also what you could do. Like looking at the leaf area, what has happened, right? We've gone from 180 to 270 to 300 to 315. So you've increased, you've selected for a larger leaf area. In terms of fruit weight, right, we've gone from 9 to 10 to 55 to 65. Again, you're selecting for a larger weight. And then for flower diameter, you've gone from 3 to 5 to 9 to 11. Again, you're selecting for a larger diameter. In terms of plant height, however, they've gone from 153 to 170 to 120 to 136, right? So it's selected against a larger plant height. They want a smaller plant height. Alrighty. I'm going to skip this one and skip this one, right? Here are the solutions that you can come back to, right? And we're going to do some Q&A before we take our next break. Alright, so again, I'll do like another 5-10 minutes of answering questions, and then we'll continue on. So I'll leave it there. Easy, so we can stop that. Alright, show Q&A. Awesome. Okay, so this seems like it's the most, the highest rated one. So, okay, I can understand the concept, but when I get to a SAC, I space out and forget information. Do you have any tips for studying/understanding content better? Good question. This is something like I get quite often from students, where it's like, oh, it just doesn't stick, right? Or in a SAC, you just blank out. And I know that happens sometimes, but I'd probably say the best way is what we call something like active learning. What this means is that, if you're studying by, let's say, just rereading your notes, or you're the way I like to think about it is if you're learning by trying to put information into your head, right, you're more likely to not remember it. Whereas if you try study by getting information out of your head, right, it sticks better. This is what I mean by that. By doing practice questions, what you're doing is you're reading an application question, right? You're reading a question and you're trying to think about, the information that's been stored in your head, right? And then you're sort of extracting that information and pulling it together to make a response, right? So you're getting the information from your head onto paper. That is a far more effective way of learning things and understanding things than just rereading your notes, which is where you're trying to get that information and put it back into your head, right? Because I can assure you that's just going to lead you to blanking out or forgetting in stressful situations. So that's one thing, doing questions is my biggest tip. Otherwise, I'd probably see what else is happening, right? So if you really do understand the concepts but then just sort of forget, right, there might be another sort of thing happening, right? It might just be the fact that, as I said, it might be a difference in study strategy that you need to do. So if you're just rereading notes and writing notes, that's different and not as effective as, doing practice questions. But it might be, so practice questions is one. It might be the other things I talked about too. So when I was talking about the wellness stuff, again, I'm not kidding. When I say about, looking after your well-being and whatnot, it's true. If you guys, I know it's so common and so, everyone maybe not everyone, it should be, hopefully it's not everyone, but, if you're like me and are going to bed late at night, not getting enough sleep, and obviously you guys have to wake up early to go to school, and not getting, a lot of sleep and, not sort of looking after yourself properly, then that's not going to let you, fully it's not going to make you go to the highest potential that you can actually reach. So it might be things like that that's happening. But I think, practice questions is probably my biggest tip. That's what worked for me. But yeah, think about other things. Is your study strategy working for you? Maybe it's not, and maybe you need a change, right? Are you sort of just studying 24/7 and that's it? Because if you're not taking breaks, then that's sort of counterproductive as well. So that's what I would say. Hopefully that answers your question. Thank you. That's nice. I really appreciate that. So let me save this question here. When should I start practice exams? When should I aim to finish the course by? And should I start practice exams even if I haven't finished the course? Good question. And I like the last bit. If so, how? Right? That's what I asked myself, right? When I saw some of my mates doing practice exams in May and, really early on, I thought what the hell? How are you doing exam questions when we haven't even finished? So, personally, and again, it's a very personal thing. Some people do what works for them. But what worked for me was, I thought I first need to finish the course. And so I waited more or less for when my school finished. As I said, I did get ahead of my school. So, during these holidays, right, these holidays, what was I going to say? During these holidays, I would try get ahead up on Area Study 1 of Unit 4 and probably a little bit of Area Study 2. And then as I go through school, while treating school as revision, I'll try finish the course whenever possible. Obviously, though, you can't get too ahead while you're doing school because you're going to have SACs coming up, right? And you've got to focus on them because that's your priority. So if you finish when your school finishes, that's okay. If you finish a few weeks earlier, that's also good. But try to finish around, it depends as well if you're year 11 or year 12. If you're year 11, I shouldn't say this, but you've got the time. If you're year 12, it's really hard to sort of manage and balance all your subjects, right? So I would probably aim for end of August. If you're going into September, that's a bit pushing it a bit, I think. But try, I think, end of August, because they also did cut out a lot of content too from Unit 4. So there's less to actually learn, and I think that should make it easier for you guys to actually finish the course earlier. Hopefully that answers it. So I would start practice exams as soon as you finish the course, so probably about August, and try to do as many as you can. I would also recommend, I think the magic number for me at least was, try to do at least 20 practice exams. It's a big ask, right? I get it. When I was in year 12, I didn't do anywhere near 20 exams per subject, right? Because I just didn't have the time. But as a year 11 student, for the year 11s out there, you've got the time. You can do it if you want to. this is a bit sad, but like when I was in year 11, I did like 40 something practice exams for bio. So that's a bit too much. Like, you don't need to do that much, right? You can still get a 50 by doing like half the amount I did. But like it's also within reason as well. Like you'll start to realize after doing exams that it's just like, yeah, I'm seeing these questions pop up again and again and again, right? And it gets to the point where just doing practice exams is just reinforcing the skills that you've gone over. All right, what YouTube channels do I recommend? Bit of a tough one. To be honest, I didn't follow any particular YouTube channels when I was doing Year 12 bio, or like when I was doing, BCE bio. I've heard from the other tutors that, we got one tutor who absolutely loves the Amoeba Sisters. I have to be honest, I really don't like Crash Course. But that's just me. I just feel like they go through it way too quickly, and it goes through one hour at the other. I don't know, for me, it's not like a particular YouTube video or YouTube channel that I follow for, studying. What I would say instead is, it depends on the topic. Like if I remember when I first did immunology and tried to learn about the adaptive immune response, that was something which the textbook didn't really explain too well. So I went to look at, the atarnotes course notes to look at the summary that they've got, which, again, a bit of a shameless plug, but like it's pretty good. But I'd also look up videos about immunity and stuff, just specific videos, and have a look at a few. Right, some of them would have been trash. Some of them would have actually been really good and was spot on and was directly answering what I was not completely understanding properly. So I don't know. I don't have any recommendations for any particular YouTube channel, but rather like the way I did it was I just looked up videos that I wanted to cover, like the topics that I wanted to cover. Good question. If you could redo 3-4 bio, what would you do differently? That's a really good question. Focus on my weaknesses. That's probably the biggest one because there's a lot has changed since I did bio compared to when I've taught bio. What I realized was when I was first teaching bio, so going over the course again, I noticed that when I was in BCE, I didn't really fully understand things like allopathic speciation, natural selection, and all that other stuff, right? And there are other concepts too, like human evolution and all that sort of stuff. I never really understood what was the expectations from VCAR. Even by doing lots of practice questions, there were some topics which I didn't really fully understand until I started actually teaching and looking more into these topics. And so for me, I would say focus on those weaknesses. So I think for me, I really loved it. I said this before, but I really loved doing Unit 3 and studying Unit 3 because it was just something I was just better at. But I didn't spend a lot of time on Unit 4. And I think on my exam, I remember what I got on my exam because I got the marks after I did it and got my study score. So I full marked the multi-choice, which sounds like, oh my God, that's what the hell? But it's actually not that hard to full mark your multi-choice. I think it's something which you should aim to do if you're looking for a 40 and above. Because the multi-choice, the answer's in front of you, right? And in bio, there's only a handful of trick multi-choice. If you know the content, you can do it. The short answers, though, they're a killer, right? The teachers are just shit when they mark, right? The VCAR examiners, they're really, really, really strict, and so you're bound to lose some marks there, right? And that's okay to lose some. So I think for me, I think I lost 9 marks in the short answer. So in total, my exam was out of 110, so I got 101 out of 110, and that got me a 46. You guys obviously have your exam out of 120, and what I would say is focus on the short answers, and particularly the sections the topics which you're not quite confident in. So for me, that was Unit 4 type questions. That's really sort of where I lost most of my marks because, again, back when I was doing when I was in Year 11, I just sort of glossed over it a little bit and didn't spend the appropriate amount of time, which I should have. All right, so that's my advice. And then I'll finish up with this one, then we'll take a break. So, how to finish bio exams on time? Good question. It's a bit of a tough one because what I found with me personally was that I struggled, really struggled to finish my SACs on time. And I remember there were maybe a handful of SACs where I didn't quite finish, or at least finish the last question or whatever it may be, because I just wrote way too much. So for me, it was about really practicing writing out short answers and doing practice short answers and refining my responses. And by looking at I've said this before, but by looking at model solutions and model answers, you sort of begin to sort of develop an understanding about what's the expectation from VCAR. And you will cut down on time by writing your answers more effectively. Again, I really hate to it might sound like a shameless plug, but I'm dead set 100% serious when I'm saying this. Another thing that really helped me was with the course notes from Shoot Smart, the bio course notes, at the end of the book, I remember there was like an exam advice or exam tips, and it sort of it had a few tips here and there about how to write your exam questions. Actually, let me just double check. Give me a second. Because I swear to God, this actually was something I looked to and I actually thought was really, really helpful. So give me 1 second. Close that. Yeah, so they sort of go through about communicating with short answers. It's not too much in detail because I think there's a bit more detail in the topic tests. But yeah, for me, this was really, really helpful. And I'm saying that from the bottom of my heart. I genuinely found this really, really useful when I was doing BC Bio. And it's something which, just getting to the point, in this, if you guys, have a look at this in your own time, I remember this paragraph saying that, to get to the point, instead of writing this, the answer is artificial passive, this is because blah, 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 you just get straight into it, right? So artificial passive, colon, or semicolon, response or explanation, right? Forget all the crap that you need to, all the unnecessary crap. You just take that all out and just write what's necessary. So yeah, I hope that answered your question. So yeah, that should cut your time on the short answer. And then for me, I feel I was always pretty quick with the multi-choice. So what I did was I told myself that, okay, I'm going to try and get the multi-choice done in the first 15-20 minutes of the exam by using my reading time to get through as many multi-choice as possible. That way, by rushing through the multi-choice, I would spend time, a lot of time on the short answer, because for me that I sort of tended to sort of write a bit more than what might have been required. So for me, I thought I wanted as much time as possible. And that way, I did end up finishing the exam I think it was about 20-30 minutes early. But then by doing that, I was able to redo all my multi-choice. I fixed up 1 or 2 mistakes I made when rushing doing it, and then went over some of my short answers. Anyway, so it's 3:30 now. I'll get back to some of these questions in a moment, but what we'll do is we're going to take a break and we'll go through some of the content. We'll continue on with the content. So in terms of the break, we'll come back at about, we'll come back at about 5-10 minutes. Let's say, 3:35. We'll start back at about 3:35, so 4 minutes. And then we'll do our last content block, which I hope to sort of get through the next sort of section of Area Study 1, which is all about the evidence for evolution. And then the rest that we don't cover, as I said, don't stress, right? I know last lecture series, I got a few comments apparently saying that people were upset that I didn't get through all the lectures here, all the slides, which, I just want to let you know that's intentional. I don't intend on going through all the content because the reason why I made the lecture slides 100 slides long was because I thought, okay, I'll probably get through 60 slides in the 3 hours that we've got, but the rest of them, I'm leaving them there for you guys to then take home and have an additional resource. So instead of just making it a 60 slide lecture and purposefully leaving out that stuff which we didn't get through, I'm leaving it in there so you guys have extra content to bring home. So hopefully you guys can appreciate that. But yeah, so that's what we're going to do. So again, don't stress out here. I just want to say one more time, if we don't finish, that's okay, right? I'm not planning on finishing all the slides because again, bio is a big subject. There's so much content in the subject that it's impossible to get through all of it, or at least half the course in 3 hours. So I'll leave you guys to it. I will go have a quick break. You guys have a quick stretch. We'll meet again in 3 minutes. So 3:35, we'll continue again with Area Study 2 of Unit 4. I ramble. I speak way too much when I should just stop speaking. All right, let's have a break. 3:35. We'll come back. Awesome. I love some of these questions. Anyway, at 4 o'clock, I'll stop and then we'll just do questions at 4 to 4:15. I'm loving some of these questions that are coming through. Anyway, so 3:35, short break. I know, I'm sorry. It was so short, but that's okay, right? Better for you guys because we get through more content. So, I just want to highlight that, what do I have to highlight? So, Shoot Smart. I just want to, so you might be aware of Shoot Smart, you might not, right? So, if you are, if you're not, that's okay. What I need to tell you guys is that with these lecture series, again, you may have heard from this from the other lectures that we're offering, again, another special promo for you guys. I'll show you what Shoot Smart is first, because otherwise it'll just make no sense what I'm talking about. But what is Shoot Smart? So TutorSmart is effectively like our tutoring sort of side of, or our tutoring company for atarnotes. So obviously atarnotes, right, to survive as a company, right, you can't just do, you can't offer everything for free as much as like we'd want to, but that's a bit unsustainable. So what do we do for TutorSmart, right? So for a very low cost, we offer sort of a weekly tutoring program, and given the COVID sort of the COVID environment, what this means is that we offer sort of like live streamed classes. So sort of like what we're doing right now, but I personally use Zoom to do it. We offer weekly 90-minute classes for particular subjects. And in addition to those weekly 90-minute classes, students in TutorSmart are able to access sort of further sort of one-on-one help with particular tutors for any subject that they're doing at school, and they also get a whole bunch of resources that come with those subjects. So what I can show you here is this, like if you did sign up to TutorSmart, this is sort of like what the student sort of portal looks like, and we might go again. Bio's not here, but let's say let's say we're a bit special and we pick. Oh, why is it signing me out for? Oh, that's annoying. Okay, give me a second. Okay, that is really annoying. All right. That's my head. There we go. All right, so let's go again. Let's say we're special and we do special math, right? So let's say we go to the current let's go differential equations part 4, right? So what you'd see is, you know, each student every week would come here, right, and watch their sort of live streamed lecture. So, you know, if you want to have a look at Natalie, right, I've never worked with Natalie, but that's okay. She probably works at one of the other centres. I work from Moonee Ponds. But there you go. They're going through sort of special stuff, all that fun things, which caused me to like hate this subject. No, I actually didn't hate special. I thought it was actually the best math out of all three. But yeah, you get the 90-minute classes plus additional resources. So for special, I guess that includes worksheets, but for bio, which I can show you on my screen, involves other things too. So, oh my god, I've got to sign in again. Mate, this technology is wrecking my promo. So slow. That's not good. Alright, let's go back. We'll try the cheeky shut off, shut on, right? So let's go back into this. Alrighty, hopefully we're not getting any problems now. Right, so we go Bio 3-4. What do we get for Bio 3-4? Okay, so where are we? We are, let's say, like our most recent week, human evolution. Right, okay, yeah, we got a PowerPoint, we've got some worksheets, we've also got Saxon exams, and so forth. Right, so that all comes with the TutorSmart program, and then other things that you guys see with this, as I said, you can get access to, you know, talking to tutors one-on-one. Just give me 1 second. Right, so what you can do here is, I think, is it in here? It's somewhere on the dashboard. But anyway, and then you can also post any of your questions on the website as well. So, for example, let's go Bio 3-4, searching questions, right? And then you can see what everyone else puts up, and see what the tutors sort of respond. So that's pretty cool too. Anyway, so that's pretty much TutorSmart. You can actually learn more about TutorSmart. I think there's going to be a session which explains more about what TutorSmart is all about next Monday, and I think it's like only 30 minutes, so it's not too long out of your day, and if you're interested, why not? Now, another thing, like Get Unlimited, right, if you use this promo code, you can get a fortnight for free. Again, why not, right? I don't get anything out of this, but like, you know, like if I was a student, I genuinely would do this too, because it's free tutoring for a fortnight. Why not snap that up? It's free. Everyone loves free things, right? I love free things, especially when you live in Brunswick, where all the cafes overcharge, like paying $20. Okay, have I ever paid $20 for like brunch? Yeah, I think I have. Yeah, that's pretty sad. But anyway, but look, it's free. Why not snap that up, right? So I just showed you a few of the things that you can do on TutorSmart. Have a look at it though yourselves, right? Yeah, there's nothing to lose, right? Again, you can opt out. Nothing gets taken from you. You know, like there's no disadvantage, right? There's only the advantage of, you know, signing up, right? And trying it out for 14 days. And if you like it, you can stay on. If you don't, that's okay too, right? It's 14 days trial. Again, you know, just be like, you can be like one of those people that just makes an account on like Apple Music or Spotify, tries it out for a month. If you don't like it, you just quit. If you like it, then you can stay on board. It's up to you. Righto. So let's continue with the content, right? So for the next 20 minutes, we're going to go over the evidence for evolution, right? Hopefully you like my memes, by the way, but anyway, I'm pretty proud of them. So in terms of evidence for evolution, the first thing we need to think about when considering, you know, evidence for evolution is fossils, right? And we've got to think, what is a fossil? It's pretty much the remnants or the remains of, or, you know, pieces of, the remains of, I guess, deceased organisms, right? And the way fossils are actually made is that the fundamental idea is that we don't want the fossils to decompose, right? And in order to avoid the decomposition of the organic matter, right, we want to make sure that they're covered, right, really, really quickly, so that they don't get too much exposure to oxygen and other sort of decomposers like fungi and whatnot. We want to make sure that they're not in, you know, really hot temperatures, and that they're not in, you know, acidic environments. So the first thing is that we want to cover them, right? That could be covering, you know, in some type of material like sand or silt or volcanic ash, right? And we want to protect them from scavengers, right? So for a fossil to form, right, we don't want scavengers messing up the decomposed body, right? You could imagine if you've got this decomposed animal right here, and you have predators or scavengers which rip apart the limbs and spread them around in the environment and all that sort of crap, it's going to be more likely to decompose. And then, as I said, we want this slow rate of decomposition, right? And that means low oxygen, alkaline conditions, so basic conditions, high pH, and we want a cold temperature. Then over time, it's going to just get further buried and buried and buried, right? And that's going to lead to the production, the generation of this fossil. Now, when looking at fossils, we can date them, right? This is a bit of a meme from Adrolo. We love Andrew Dash, right? He's fantastic. But looking at dating fossils, right? This is not what we're talking about. When we talk about dating fossils, we're thinking about how old is the fossil. And there are two ways to date fossils, right? We can figure out their relative age. So that means figuring out, is fossil X older or younger than fossil Y? It doesn't tell me exactly how old they are. It just tells me how old is one fossil relative to another fossil. Is it older or younger? The absolute age is telling us, yep, this fossil is X amount of years old, right? So, I don't know, it might be that this fossil is 5 million years old. So it's the specific age of the fossil. Now, when we're talking about relative age, right, how does this actually work? It's based on like the basic principle that, okay, when we're thinking about mountain ranges and, any sort of like mountainous sort of formations, right, and rock formations, that obviously, to form like a big sort of pile of, sort of mountain or hill or whatever it may be, you've got to start from the ground up, right? So like dirt or rock or whatever the substance is, piles on on top of each other, on top of each other, on top of each other, and so that means that the most bottom layer is the oldest layers, whereas the most, the highest layers are the most young. Right, that makes sense. Hopefully, right. You don't obviously you don't start with like dirt just floating in midair, and the dirt just like sprinkles down and sort of fills up the cavity. Right, that just doesn't make sense because of gravity. Right, we start from the bottom and work our way up. As a result, what this means is that if we find a fossil in this rock strata versus this rock strata, say this is fossil X and this is, and let's say this is fossil A, what this means is that we can say that obviously this rock strata is, or stratum is older than this rock stratum, so fossil A must be older than fossil X. Hopefully that makes sense. This also brings us to this idea of indicator or index fossils. So this sort of means that, when we're looking at two different mountain ranges, so looking at location 1 versus location 2, we can pick out sort of a fossil that only lives sort of a short amount of time and sort of compare that, okay, how old is that, I'll give you an example. The best example I could probably show you is looking at this guy over here, right? Is that the best example I can give you? Yeah. Alright. I might give you actually this example here. So looking at this like shell type structure, right? This is a fossil found in layer C, right? Note though that this has been conveniently labeled and color coded for you so that you got like brown, brown, brown. In real life, you don't have this nice convenient color coding, right? The rock is just like, similar colors and whatnot. So you can't just color code, right? This isn't real life. So what happens in real life is that you look at this index fossil and you say, okay, what fossils are around it, right? Or actually you look at sort of where this fossil is in the different rock strata of the different locations. So location 1, it's sort of in this like second top layer. In location 2, it's in the most highest or top layer. In location 3, it's not even present. And in location 4, again, it's in the second top layer. So in that way, because this fossil which lived a short amount of time, right, is found in relatively the same, which was because it lived in the sort of short amount of time and is found in all three of these locations. It then means that the fossils around it in the same sort of layers of the rock are similar ages. So it means that everything in this rock strata would be of a similar age to everything in this rock strata. And likewise, that's similar to everything in this rock strata. Hopefully that makes sense. When we're talking about finding the absolute age of things, right, so the the defined age, so whether it's 1 million years old, 2 million years old, not just younger or older, right, what we do is we use isotopes, right? I'm not going to go into the specifics of isotopes because no one likes chemistry, but what this is mainly talking about is when we've got sort of like an unstable molecule, right, a radioisotope, and we're looking at how much it has decayed over time. So to give you a specific example, carbon dating is probably the most common one that you see in VCAR exams. In this situation here, we're looking at the proportion of carbon-14 to nitrogen-14. So carbon-14 is a radioisotope that's really unstable, right? And over time, it will break down into nitrogen-14. For 50%, or the time it takes for 50% of carbon-14 to break down into nitrogen-14, that time is what we call the half-life, and specifically it's about 6,000 years old, right? Or it takes about 5,730 years. As a result, this means that if I find some, a fossil, this is a good example, right? So we've got this, this organism which will eventually die, right? And at death, right, straight away, it will have 100% carbon-14. But then you could imagine that every 5,730 years, this amount gets decreased by half. So we then will record 50% carbon-14. 25, 12.5, and so forth. So then what this means is that if I figure out that there is a 6.25%, right, if I give you a question and say, a fossil was shown to have, 6.25% of carbon-14, how old is that fossil, right? I don't think they would ever give you like a mathsy question like this, but this is just using maths, right? Because what's happening here is that every half, right, you add 5,730. Obviously, this is a bit of a weird number, so I'm just going to say 6K, like 6,000. So, right, what this means is if I found a fossil that has 6.25% carbon-14 remaining, this means that the fossil is 6, 12, 18, 24, about 24,000 years old, a bit less. Does that make sense? Hopefully it does, right? And you see this by this graph too. Right, so every 50%, right, is about 6,000 years old. The interesting thing about carbon dating, though, is that because the half-life is relatively small, right, 6,000 years obviously sounds like a long time for us, but when you consider that fossils might be millions and millions or billions of years old, right, it's not that long of a time. So what this means is that, because you're dividing it half every single time, like every 6,000 years, the amount is halving. So just to reiterate what I'm saying, so you've got, 100% to begin with, then at 50%, there's been 6,000 years, I'll just say 6. Then 25% will be 12, 12.5 is 6, 12, 18, 6.25 is 6, 12, 18, 24, and so forth. And you will see that soon you'll start going to get, 0.0 something something something, 0.00000 something something, right? The numbers are going to get so incredibly small that it's undetectable, right? That you can no longer trace the amount of carbon-14 left. This sort of creates this almost like an expiry date, right? And that is about 50,000 to 60,000 years old. So once you get to about 50 to 60,000 years old, it starts to get the amount of carbon-14 left is so small that it's really undetectable. And that's why you can only use, and this is something which is sometimes tested in a multi-choice, you can only use carbon-14 dating if the fossil is young, or relatively young, so like less than 50 to 60,000 years old. Now, the same sort of principle applies with potassium argon dating, so I'm just going to skip to the next topic, but I can let you read that in your own time, of course. Now, other evidence for evolution. So aside from, the whole point of just telling you about carbon-14, you can look at potassium argon dating as well. The whole idea, and as well as relative dating, is that this allows us to build some type of a timeline, right? By having, a bit of a timeline to identify, the age of fossils in different years, right? This gives us a bit of an idea about how things have evolved over time. And this will make a bit more sense as we go through some other pieces of evidence. So the idea of dating, just to summarize, is to give us a bit of an understanding of the evolutionary timeline that has occurred over the lifespan of living things on Earth. So, in addition to this, right, we've also got structural anatomy and being able to compare the anatomy of different species. So what we find, which is really interesting, is between some species, we'll find what we call a homologous structure. This effectively means that the fundamental makeup of these, whether it's a limb or whatever the structure may be, it's sort of conserved, or it's similar to other other structures found in different organisms or different species, but they elicit different functions. So, for instance, right, the human arm, right, when you look at the bone structure, that is very similar and is homologous to the bone structure of a whale's fin. The thing is, though, hopefully you can understand that a whale's fin completes a different, entirely different sort of function to a human arm, right? A human arm can bend, it's used to, the hand is to pick up things, it's to do other things as well, right? Whereas a whale's fin is to swim, right? It's a bit different. So it's a different function, but the same structure. Analogous structures are ones which it's the opposite. It's where you have the same function, so in this case, wings are there to fly, right? But the structure of the wing is actually different. Now, it's a bit confusing about how much you need to know about this concept, because an additional factor I can tell you is that when we talk about analogous structures, this shows us that these species aren't necessarily closely related, but they live in similar environments and have suffered the same selection pressures, meaning that they had the same selection pressures which provided some selective advantage to develop wings. However, when we talk about homologous structures, these are structures which, these are from species which are closely related, but, don't have the same selection pressures, and therefore that's why they perform different functions. The reason why I say it's a bit confusing to know how much you need to know about this is because of the changes to the study design given, COVID-19. So that point that I've just told you is then related to something we call convergent and divergent evolution, but that has been taken out of the study design. So make sure you know what I put onto this slide. The second point I told you about how this relates to evolution, I think you should know too, but I just didn't want to have it here just in case that's something which VCAR associates with the stuff that they took out with convergent and divergent evolution. Now, this is probably my favourite evidence because it looks cool. It's what we call transitional fossils, or transition fossils. So, as you could imagine with the timeline of evolution, you could assume that, one organism doesn't just randomly evolve to form another organism, right? Like a fish didn't just one day wake up and form a reptile, right? There would have been a bit of a process or a series of mutations and a series of generations which led to a transition from species Y to species X, for example. And it's what we call transition fossils. It's these fossils, or the remains of these organisms or these species, which sort of indicate that transition from one type of species to another. And what you're seeing in the image here is a transition fossil showing you sort of the intermediate species of a reptile and a bird. So what's predicted based on the age of fossils is that reptiles eventually went to form birds. or at least some reptiles went to form birds. So birds are sort of closely related to reptiles. And this is sort of evidence to also support that, to show a transition between the two, right? So this is, as you can see, it sort of looks like a reptile when you consider just the body of the organism, but then you've also got this like wing structure as well. So that also indicates its ability to potentially fly. Another thing is what we call vestigial structures. These are structures which literally have no function, right? They're pretty, they're not really important, but they're remnants of features which are shown or found in similar similar organisms. Related similar organisms. When I say similar, I don't mean like a dolphin and a shark. I'm talking about like a whale and a human, right? Which are evolutionarily, if that's the word. Related based on evolution, that's what I'm trying to say. Anyway, so vestigial structures. So the classic textbook example is, I think this is a pelvis and femur, which are bones found in other types of animals such as us, right? We've got a pelvis, the pelvic bone, and the femur, right? The bone in your upper leg, your thigh. And whales have these, but it's really, really small, and it pretty much does nothing. Right? So it's got no function, but the reason why it's there, it's predicted that it's there because it's sort of evidence to show that whales have descended, or at least not descended, but are in some shape or form, similar to other organisms which share this structure. A modern day example, and you can test this out yourself, is if you extend your arm out, like you can see in the photo, and pinch your thumb and pinky together, and then sort of flex your hand towards you, you might see I'm not sure if you can see this, but you might see like this sort of like strand or this tendon sort of appear, right? You can see it in the image here, right? You might see a tendon appear, or you might not. Fun fact, if you do not have the tendon, you're a minority in the population, which is apparently more evolved than everyone else. So it's a bit of a flex if you're a part of that subset of the population. And it's assumed that because what that tendon is, it's a part of the muscle called palmaris longus, right? This is just a fun fact. And that's a muscle which is shown in some of our hominoid sort of relatives, right? So things like apes and whatnot. And it's used to, I'm pretty sure it's like useful when climbing trees and other sort of stuff, which, apes and whatnot do. But because humans don't really need to do that anymore, right? It's no longer a selection pressure to determine, how well you can swing from one tree to another, right? That's a structure which has been evolved out, right? And yeah, so that's apparently more evolved if you don't have that structure. Anyway, that's just a fun fact. Just the next sort of section, I know I'm being cautious of time. I might just quickly go through this because it's really quick. The last sort of section to this is sort of looking at the molecular evidence of evolution and seeing how we're related based on, the molecular basis, or on a molecular basis. So you may have heard like really random statistics like humans and bananas are related by 60%. Anything like, what the hell? Like, how am I 60% related to a banana? How those sort of figures come up is by sequencing DNA or proteins. I'll tell you what that means in a second. So, DNA sequencing. This is the process by which you take a gene and you sort of figure out the exact nucleotide sequence of the gene. So it might be, a gene A is like A, T, C, G, T, etc., 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 right? So this is the DNA sequence. And what you do to determine the similarity between two different species is you take the related gene or the similar gene. So it might be you look at the gene that produces cytochrome C, for example. You might remember cytochrome C as being a major factor in apoptosis. So you might take cytochrome C protein and you might look at the gene from a human cell and from a chimpanzee cell. And what you do is you identify how many differences there are in the DNA. What you can hopefully appreciate is that the more differences there are in the DNA, that assumes that there has been a longer amount of time since the two species has, evolved from each other, right? Or since they've shared a common ancestor. Because what it suggests is that the longer, the more amount of mutations means that, to have more mutations present would require more time. You need like a longer period of time to develop mutations, right? Like if I give you 1 million years, like if someone lived for 1 million years, right, they would objectively develop more mutations than someone who lived 1 day, right? So the longer amount of time between, the separation or the divergence of a common ancestor would lead to the accumulation of more differences in the DNA, the accumulation of more mutations. And this is shown here. So what we're seeing here is a table where we're looking at a few different species, and we're looking at the differences in the nucleotide sequence for the cytochrome C gene from a human cell. And what we can see is that we are technically more related to a rat compared to a chicken or a yeast cell, right? Based on the fact that there are only 34 differences in the rat cell, compared to 110 differences between the human and yeast cell. The same principle applies for amino acid sequencing. Here you're looking at the sequence of amino acids, right, of equivalent proteins, of course, and that way the more differences in amino acids, the more differences in amino acids, the less related the two species will be, right? Because it indicates that the common ancestor is a bit older. And this is also shown here, right? So when comparing, pig, cow, and human insulin, right, the protein, you can note that, for at position 30, the pig and cow have a different amino acid than the human beta chain of human insulin, for instance. So again, the more differences means that the less related they are. And to summarize all of that, we can then visually put this on paper, right, by creating what we call a phylogenetic tree. And what this is just sort of like a visual representation of how related species are, whereby what you see on the right-hand side is all the modern-day species, and all these little intersections here represent ancestors. This one, which I'm circling right now, that is the ancestor species, or the most recent ancestor species for species A and species B. Then, this one over here is the most recent ancestor for, I guess, like all of them, right? And then I guess the key thing to understand is that, how do we know which ones are the most related? Well, the more, recent the recent ancestor is, or the common ancestor is, the more related they are. So, for example, and the way you can sort of do this is by tracing backwards. So, to see how related A and B are, you go look, and they intersect here, at this point. If I wanted to see how common A and C are, you would trace back, and they intersect here. And what you can see is that the green is older than the red. Because of that, because obviously this is like time, the x-axis is sort of like time, where this is old, this is, we go from old to young, right, or most recent. And the more recent it is, obviously the more related these species are going to be. Hopefully that makes sense. Alrighty, in terms of content, I might leave it there. Give me 1 second. So, in terms of content, I might leave it there. What we didn't go through today, and don't stress because again, you've got the slides, which hopefully help, is human evolution. There are some good memes too, by the way. But anyway, human evolution, I've got some questions that you can have a go at. And then, so we continue with human evolution. And then I quickly put just a summary of Area Study 2, if you want to have a look into that as well. So, before we finish up, I want to have a look at some of these questions and spend the remaining amount of time that we've got by going through, obviously the questions you want to ask me, because I think that's probably the best thing you guys can get out of this. Again, we've got 223 questions waiting, so it's a bit unlikely we'll get through all of them, but if you want to upvote, if you guys look through some of these questions, feel free to upvote any that you think there's, questions which which I haven't answered yet, or I'll put any which you want to be answered and which that you want to be answered. I think that's all I have to say. Cool. Right, and we'll do this till about like 4:20. So in terms of this question here, what percentages were you getting in your SACs to get a raw score of 46? Okay, this question, it doesn't really matter. I mean, look, I was getting, for Unit 3, I was getting about like 93. I think that was my average from memory. It was like low 90s, somewhere around that. And then Unit 4, I think was either like 89 or 91. Can't exactly remember. But the idea is that, again, it depends on don't worry about the SAC marks you're getting. Think about the rank that you are at your school. So for me, I could have been getting 70% on my SACs, but if I was in rank 1, then that's okay, because it might suggest that my SACs are harder than average, right? Technically, like, my SACs were fine, because in terms of the scaling of like my marks, I noticed, like, because I think when you get your results, you can pay like $5 or $10, something pretty cheap to VCAR to get your like actual data and marks for your SACs and stuff. And I think mine went up from like 93 to 97 and like 89 or 91 to like 95. And that sort of shows me that like my school's SACs were a bit harder than average, and that's why my SACs scaled up. So just be aware that it depends on your the SAC difficulty of your school, as well as the cohort strength of your school, as well as your rank in your school. So don't stress too much about if you're getting 80% or 90% or 70% or 40%, right? It's okay. Just make sure that you are doing well and are doing lots of practice questions stuff, because you want to smash the exam. That's really the key. Is it okay to cry over bio? Because I think I'm crying too much. I love how this one was approved by my boss. So like behind the scenes, one of my managers is sort of like going through and approving the questions to make sure like inappropriate questions are going up. But that's okay. If you want to approve this, is okay. You shouldn't be crying over bio. Make sure that you're worried about your well-being. That's sad. But look, it happens. I mean, I don't know. Maybe it's because I'm a guy. Like maybe I don't know if guys cry like as much. Like I've heard this more often from like some like the female students that I've taught. And the first few times I heard this, I was a bit shocked, because like going to an all-boys school is a bit like, what the hell? Like, I probably shouldn't say this, but I was a bit like toughen up, like deal with it. You didn't do well. Like we all don't do well in the SAC. I didn't do well in some of my SACs. It's okay. And I was a bit shocked. I'm like, how do you cry after a SAC? But then I've actually heard this quite often. And I don't know. I'm not sure whether it was just my cohort that wasn't like as emotional. Maybe it's toxic masculinity. Who knows? But anyway, is it okay to cry over bio? Yeah, I guess. If you feel like you need to get the feels out, just do it, right? You'll be okay afterwards. You get it out of your system. Anyway, so, but make sure make sure you take care of yourself too. If it's okay. At the end of the day, guys, right? Don't stress. The ATAR is just a number, right? I know I'm working for a company called atarnotes, right? But the ATAR is just a number. Like, don't even stress about it. Like, I think there is so much pressure, too much pressure put onto the ATAR, and people don't realise that there are other sort of avenues and stuff that like you can pursue. So if you don't get the particular ATAR that you're looking for, or the particular study skill that you're looking for, like you don't really have to stress too much, because like there are so many alternative options and pathways when you're in uni. It's actually unreal, and I know it's a bit rich for me to say, because like I was in you're probably thinking, Michael, what are you talking about? I just want to do as well as I can, which is good. But I just want to do as well as I can to get into my course straight away, and I don't want to have the headaches of having to do all that sort of crap. The thing is, once you're actually in uni, you sort of start to realise how flexible it is, and it's completely different to high school. So many of my friends have switched degrees, have taken gap years, have switched majors. One of the people I work with has like switched their majors for like 3 times, I think. I've got a friend who didn't fulfill the methods prereq and wanted to do commerce, so he just did arts for a semester, which is like 3 months, and then switched over into commerce. So don't stress too much, right? It really isn't that significant, and quite frankly, no one cares what ATAR, like genuinely, no one cares what ATAR you got after you leave BCE. I remember, like, after I got my ATAR, like, I was happy for about like maybe 10 seconds, and then I realised, that's over, that's done. He's like, let's say hello to the next chapter of my life. Like, it's a bit irrelevant. So don't stress too much. There's a bit too much pressure, I think, on the VCE. So chill. If you don't do as well as you expect, it's okay to be upset. Like, I'm not saying, you shouldn't be upset at all. Like, it's okay. It's okay. Like, don't stress too much. Like, it's going to be fine. If you want to get somewhere, right, you'll get there in the end. It's going to work out. Start my YouTube. Look, I'll put it through to my boss. I'll see if he's I'll see if she's keen on start making me start up my YouTube channel, and then if she is, then we'll start up a YouTube channel. How do I prepare for Unit 4 SACs? Alrighty, how do I prepare for Unit 4? Questions, practice questions, guys. I say it all the time, but I really like it's my favourite thing to say. Right? Practice questions. That's what I'd say, right? That's my biggest advice. I try to get through as many of these questions as possible. That's why I'm going to give a bit shorter answers and not ramble as much. When should I start doing full VCAR exams? Would it be a bad idea to start doing the old VCAR exam now? Not really. It's not a bad idea at all. It's not a bad idea at all. I would just say, just be mindful of the differences in, Unit 3 back then versus Unit 3 now. So, for example, when you go through some of those questions, you won't see any transcription translation, but you'll see transcription and translation, in your Unit 3. So there have been stuff there's stuff that has been switched over or shifted across the course as the years have as the study designs have updated. So just be mindful of that. But no, it's definitely not a bad idea to start, Unit 3 questions. I mean, look, if you've got the discipline to actually do full, VCAR, Unit 3 exams, that's great. But I'm going to be realistic here. I'd be way too lazy to do that. Because I'm a bit of a lazy person. So what I would recommend is, if you're a bit like me and you just want to do something which is a bit more manageable, set aside like an hour a week or something and do some like atarnotes topic tests or checkpoints or whatever sort of resource you have, which has practice questions that you can use. That's sort of my advice, right? And that way it's a bit more manageable and it's a bit more targeted as well, because you can then sort of make sure that, okay, I want to study immunology today, or okay, I want to study self-signaling today, right? So it's not just doing a random